Welcome, everyone, to day 2 of the 2022 Tilden Fungus Fair. I'm your host, Naturalist Trent with East Bay Regional Parks, broadcasting live from the Tilden Nature Area in Berkeley, California. Let me know in the comments where you're watching today's program from while I queue up a quick outline of the program. While I'm doing that, I'm going to tell you our opening slideshow was from local photographer Alison Pollack. You can check out her work over on Instagram at Marin_mushrooms. Right, everyone in the chat just down the hill, Berkeley Flats. Thanks for watching. Mila, San Rafael. Hey, Patrick, how's it going? Pleasant Hill, Coastal Massachusetts. Jan, that sounds really cold. Thanks for tuning in today. It's pretty warm here in California, but I did have to turn the heat on when I was driving to work this morning, so I feel you, sort of. And let's see what's going on over on Facebook. San Francisco, hello neighbor. San Mateo, Humboldt County. Crab Cove Visitor Center, hi Pia, thanks for watching. And Lake County, Blue Lakes. I don't know where Lake County, Sharon, is that? Sounds watery. Thanks for tuning in today. Remember, folks, if you have questions or comments at any point during the program for our presenters or for me, type them into the comments on whichever platform you're watching, and we'll get to them as quickly as possible. Well, it's my pleasure to bring in our first guest for the day. James Morris is a graduate student in Dr. Brian Perry's lab at Cal State East Bay, pursuing a master's degree in biological sciences. He received his BS in environmental science from St. Mary's College of California, where his interest in mycology grew in his undergraduate research lab. He's currently studying the decompositional abilities of macrofungi associated with eucalyptus trees in the East Bay Regional Park District. James, thanks for being here.
Hi, thank you for having me. Thanks for that introduction. And thank you for the opportunity to present at this year's Fungus Fair. I'm super excited.
Sure thing. Hey, so I understand you're a park ranger in Marin County. I don't know if you know this, but I'm a park ranger too. Tell me more. What do you do over there?
Yeah, so I just recently started as a park ranger in Marin County. I'm at Paradise Beach Park, and so it's a lot of like park maintenance type of work, but really doing everything: irrigation, plant management, interacting with the public is really the biggest part of it. But yeah, Paradise Beach Park in Tiburon is where I'm stationed at.
Okay. I have to say, as someone who's seriously considered going back to grad school for mycology, I have to ask, how do you balance working as a park ranger and going to grad school?
It's hard. It's a lot of work. Yeah, it's difficult. I think just prioritizing, scheduling things out so then I have time for my grad school work. And I've been really fortunate with my job where they've allowed me to have the type of schedule that can work around having a class schedule and doing lab work, working on my project, while also having a full-time job. So it's difficult. It's a lot of work, but I'm also super fortunate. So I have to, try hard to take advantage of those opportunities for sure.
Yeah, well, sounds impressive. I might want to trade notes with you after today's talk. But I'm gonna turn it over to Ranger James, everyone, with Eucalyptus globulus wood degrading ability of California native rot fungi.
Can you see my screen okay here? Yeah, I think so. Okay. So if you don't know me, my name is James Morris. Like Trent said, I'm in the Perry lab at Cal State East Bay. I'm studying biological sciences. And Dr. Perry will be presenting later on today, so please stick around for that. And I want to give you a brief introduction to sort of what inspired the project and why I'm studying these wood-degrading fungi. With response to the increased severity of California wildfires, East Bay Regional Parks District has this wildfire hazard reduction and resource management plan. And part of that plan includes reducing fuel loads by thinning invasive plants like eucalyptus trees. And by thinning, I mean they cut down the trees and they'll leave the stumps because they're very hard and very expensive to remove. And then they treat the stumps with herbicides to prevent resprouting. So eucalyptus very readily will resprout from those stumps. And so they treat it with this herbicide, Garland 4 Ultra. And other jurisdictions, like in Marin County, thin invasive plants in similar ways, leaving the stumps, and then they'll use really the same herbicide, Garland 4 Ultra, to prevent them resprouting. So I've been a part of projects where we go out thinning invasive plants and then people come in afterwards spraying this herbicide. And that could have detrimental effects on the environment, especially if you're doing this in a watershed, right? This herbicide leaking into the water supply and can affect all of the aquatic taxa. So this project, well, first, let me give you a little bit more background on Eucalyptus globulus. That's also known as the blue gum eucalyptus, and that's what you see all over the East Bay Hills. The blue gum eucalyptus is allelopathic, so that means that the leaf litter and stem flow release toxic leachates into the soil, and that suppresses understory growth. So by stem flow, I mean, if you've ever seen eucalyptus after the rain, there's that kind of foamy stuff that comes down, and you'll see it at the base of the tree too. That has these toxic chemicals that will actually suppress the growth of plants around them. And that results in these really huge eucalyptus monocultures that you see in the East Bay Hills, where it's just like only eucalyptus trees, and it's kind of taken over the native oak woodlands in a lot of areas. These trees are really difficult to remove, especially the stumps, and they'll readily resprout, like you can see in the picture on the right here. And past studies have used surveys of native fungi to isolate as potential biocontrol agents. So these fungi that will eat and degrade wood, using those as agents rather than using herbicides to prevent eucalyptus resprouts. And so that's where my project really comes in for the East Bay Regional Parks District. The project overview and the overview really of this presentation is a survey of the fungi in the East Bay Regional Parks District. And then I take those fungi that I survey and collect and use some laboratory-based growth and degradation assays. And I'll use the data that I collect from those assays for further studies on biocontrol, right? So really the point that I want to emphasize here is that instead of using herbicides for biocontrol, perhaps we can use naturally occurring fungi, wood decaying fungi, to prevent resprouts and to control invasive species. And this can be used, across multiple jurisdictions in the East Bay Regional Parks District, and hopefully, maybe I could bring it to Marin County Parks as well. So now I want to go over my project a little bit. Here's one of my survey sites, the Blue Gum Picnic Area in Tilden Regional Park. You can see just all eucalyptus everywhere. And when I'm surveying, I'll go out often in times of like extreme rain, like that huge rainstorm we had a few months ago. This is my girlfriend, my mushroom hunting partner. We'll go out to survey sites like the Blue Gum Picnic Area and basically walk around, look for mushrooms on stumps, on logs, on eucalyptus trees. And then I'll identify those mushrooms using their macromorphological features or using like a dichotomous key in a book, and then collecting tissue samples for lab culturing and identification by DNA sequencing. And so what I mean by that is I'll take mushrooms off of the tree, I'll put it in my little fishing tackle box, I'll take it back home, put it in the refrigerator, and then the next day I'll take it to the lab where I can put it in culture. So this is like a really fun part of the project, going out into the parks, looking for mushrooms on trees, trying to identify them. And so it adds like an extra level to just going out and hiking as I'm surveying for mushrooms on all of these trees. Here are my survey results for this winter, at least. I found 9 species. And I'm not going to go into all of them, but I found multiples for some of them, and that's where I'm doing my growth experiments and my degradation experiments. But really, after that big rainstorm that you saw in that previous picture, we went out and we saw, literally thousands of Sterium hirsutum growing on these logs. This is also called the hairy curtain crust, but literally thousands growing all over these downed eucalyptus logs, all over these stumps, thousands and thousands just right immediately after that first big initial rainstorm. So I thought this could be a really good potential biocontrol agent just because of its prevalence in the natural environment already. And then after I collect those mushrooms and put them in a tackle box and bring them home, I'll bring them to the lab to culture them. And if you haven't done culturing in the laboratory before, it's like on a little petri dish, and I'll use malt enhanced agarose media. So it's just a gel media. Malt enhanced means I'm just adding extra sugar so the mushroom has food. And I add an antibiotic because when you take mushrooms from the field, there can definitely be bacteria on there. So I want to make sure that what I'm growing is a fungal growth and not just bacterial. I'll take a tissue sample from that mushroom, put it on my media plate, and then I'll incubate it and see what happens. And if I get a pure culture, then I've created what's called a eucalyptus sawdust-based media. So I take eucalyptus wood and literally just chainsaw it into a sawdust, and take that sawdust, put it in a bigger petri dish with agarose to create this matrix. And then I'll take some of the mycelium from that initial culture, put it into the center of that dish, incubate it, and see how well the fungi will grow on a eucalyptus sawdust-based media. So will it grow on this sawdust in a lab setting? And I'm doing that for my growth experiments. So the growth experiments are kind of two-part. The objective is to measure the mycelial growth on my eucalyptus sawdust media in 2 parts: mycelial density. So on a 6-point scale, measuring mycelial density. You'll see what I mean in just a second, and you'll get to see the eucalyptus sawdust media plates as well. And then taking weekly diameter measurements to see how quickly and how far the mycelium will grow. So here are those eucalyptus sawdust media plates. You can see here, those are all just eucalyptus wood chips that I've chainsawed from logs. And I use this point rating scale to quantify mycelial density. It can be kind of subjective, but the 6-point rating scale kind of gives you an idea of how we're quantifying the density here. So 0 being no growth, 5 being extremely dense. And I'll zoom in here so you can see what I mean. On the left here, I would rate that a 5. On the right, I don't know if you can see the mycelium there, but I would rate that a 1, just to give you sort of an idea of what could be kind of a subjective scale and how I'm rating that there. And with that, I also did those weekly diameter measurements. So just taking calipers on those plates and measuring the diameter and taking averages for each of the replicates that I have for each taxa. And here are the results for the density, right? So on the y-axis, we have average mycelial density. On the x-axis, we have my species. Again, I only did 5 out of the 9 because I had multiple replicates for those. And you can see Stereum hirsutum has the highest mycelial density of 4.13 average. Polyborus mcmurphyi had the lowest density of a 1.0 average. Here are the results for my average mycelial growth on Eucalyptus globulus sawdust media. So these are the mycelial diameter measurements. On the y-axis here, you have the diameter in millimeters. On the x-axis, you have week 1 through week 6. And then you can see the different taxa color-coded. And one of the issues that I'm running into here is that if the mycelium covers the entire plate, then it might appear that the growth is stagnating. Like, let's look at Trametes hirsuta here in green, for example. Once it reaches 85 millimeters, which is the maximum diameter of my Petri dish, then it looks like the growth is stagnating, where in reality, it just doesn't have anywhere else to grow. It filled up the plate. Same thing with Stereum hirsutum here. We had some individuals that just filled up the plate. And so it appears that like this growth is stagnating, but really it's just there's not anywhere for it to go. And so to kind of supplement this graph and my data, I'm taking just mycelial diameter after just 1 week of initial inoculation. So I'll put it onto that sawdust media plate and then measure the diameter 1 week afterwards. And these are the results for that. With, again, Stereum hirsutum having the highest diameter after 1 week. And this gives like some initial growth rate data that can supplement the data that I have here. The next steps for my research, I want to complete my growth experiments. So I had, 5 of the taxa, or 4 in some cases, out of the 9 that I found this winter. I want to complete those with the others with multiple replicates. And then I want to run degradation experiment. This is, where it gets a little more exciting. Is I'll take a block of eucalyptus wood with a known mass, and then place it in a mycelial mat. So just fungal growth on eucalyptus wood chips. Once the wood chips are completely colonized by the mycelium, I'll place this eucalyptus block in there, and then see what happens. See if they'll start eating and degrading the wood. And then after a set period of time, I'll take a final mass measurement. So I'll have an initial mass and a final mass to see how much did the fungus degrade the wood. And I think that that's the most applicable to future field studies. I also want to confirm my identifications, right? So I'll identify the mushroom by its macromorphological features, but I want to verify those identifications by amplifying the ITS region and DNA sequencing. So I want to verify my identification with genetics. And then finally, further studies on the potential for fungi as biocontrol in the field. So the people over at Bay Area Applied Mycology and Hart Singer, especially Hart is another student in Dr. Perry's lab. He's doing these studies inoculating eucalyptus stumps with a fungus and seeing what happens. How does it resprout? Is it degrading the stump? But my growth experiments and degradation experiments can kind of form a scaffold for these future studies with, how well do these fungi grow on eucalyptus? And then how well do they degrade a block of eucalyptus, which is simulating like a eucalyptus stump, right? And so I can create like a sort of rating system for which fungi might be the best potential biocontrol agents for future field studies. And I really want to emphasize that these wood decay fungi, they could have the potential in multiple jurisdictions to, instead of using herbicides for biocontrol for invasive species, maybe we could use native rot fungi. And I think that's a really exciting sort of potential for the future. There is to use native fungi rather than using herbicides for biocontrol. And with that, I'd like to, again, thank you, Trent, for inviting me to speak at the Tilden Fungus Fair. I want to thank my advisor, Dr. Perry, for this opportunity. I want to thank East Bay Regional Parks District, Bay Area Applied Mycology, and really everyone else who's helped along the way. And with that, I will take any questions.
Awesome. Thanks, James. That's it sounds like you're doing really cool work, and right here in Tilden, literally, less than an eighth of a mile from my office. How have we not met yet? I'm so curious. Folks, if you have questions for James, go ahead and type them into the comments. There are questions coming in right now, but since I'm the host, I'm going to steal the first question and ask, I'm curious, how familiar were you with your target species before you started this project? Did you have to learn some of these species in the field as you went, or did you have a good idea what you were going to be seeing before you started?
No, I had no idea. I'm still, I'm not an expert in identifying mushrooms at all. I didn't really know what I was going to find. I was expecting, just looking through field guides, looking through, mushrooms of the Redwood Coast or California mushrooms, those books, what I could sort of expect to see with rot fungi that degrade hardwoods. So things that are degrading, oaks or other hardwoods, you would likely find on eucalyptus as well. But I had no idea. I'm still, very much an amateur with identifying them in the field. So I didn't have any expectations really.
All right. Well, that's interesting to know. I think based on some conversations we had yesterday, a lot of what I would consider professional mycologists just are very humble in their identification skills, and I think there's just a lot of interesting potential for collaboration between amateur mycologists and the professional and academic community. We've got quite a few other questions for you here. Mila asks, is there a particular reason you didn't include Laetoporus, the western sulfur shelf, or chicken of the woods, as some people commonly call it, since that's a known eucalyptus wood rotting fungi also?
Yeah, that's a really good question. There is a particular reason I didn't include it. There are a couple of reasons, actually. I wanted to find what is naturally occurring in the East Bay Regional Parks District, and I haven't found any on eucalyptus in the parks in my surveys. But the more important reason is that Laetiporus gilbertsonii, which is the species that's associated with hardwoods, it's a parasite, so it parasitizes plants. And if we were to use that in field studies, inoculating a bunch of eucalyptus with a fungal parasite, it could spread to our native oak woodlands as well and parasitize those. So I want to use sap probes, things that will decay dead wood and not living wood, because the potential for a parasite to spread to our native oak woodlands, that's not something that I really want to take on. So yeah, that's a really good question, and there is a reason, because it's parasitic.
Okay, well, great reason, in my opinion. Laetoporus is a beautiful fungus, and a lot of people enjoy finding it out there. But yeah, good to remember that it is a little bit more on the parasitic side than the saprobic side. James asks, what temperature and humidity do you use in your tests?
That's a good question. I had them in an incubator, and I don't know the temperature off the top of my head, but humidity, we don't account for that. That would be a good thing to keep standardized. But yeah, we keep them in an incubator, so at a warm temperature above room temp.
All right. Mitch asks, could the wood degrading fungi have unintended effects on other tree species, which I think you actually answered a little bit when you answered Laetoporus.
Yeah, it just depends on the ecology of the particular species. So if it's a parasitic fungi, then yeah, it could have some detrimental effects on other tree species. But if it's a saprobe, if it's decaying dead wood, then it shouldn't spread to our oaks or other native tree species.
Right. Makes sense. There's a couple more questions for you. However, James, your video is really laggy, so I'm just going to switch your feed off and back on. Bear with me, viewers. Let's see if this works.
Hello, hello.
Not so much. At least your voice is coming through clear.
Okay, good.
Santa Barbara. Hi, Julia. Let's see. Over on Facebook, Pia asks, this research is wonderful. Do you envision the fungi being available commercially for gardeners, etc.?
I don't know. There's not really any plans for that. This is like I said earlier, kind of like a scaffold maybe for future wood decay studies. I haven't really thought about like a commercial aspect for wood decay fungi. I'd be interested in like what kind of ideas you would have for that, but no, I haven't really thought about the commercial aspect at all, to be honest.
Well, I think there's a lot of other people out there, particularly Mr. Paul Stamets, who would jump on the opportunity to sell any type of fungi you could. So maybe you could just, sell your research to him when you're all done. Sorry, folks. Let's see. More questions here. I'm going to mess this name up. Sativivek asks, what are the byproducts? Just the growth of the type of fungus, anything else? So I assume they're referring to like fungal metabolic byproducts in the lab setting, but you're just looking at kind of the growth rates and the consumption rates, right?
I'm just looking at the growth rates and the consumption rates. As far as like metabolic byproducts, I think stick around for Dr. Perry's talk later on some fungal metabolic byproducts of these rot fungi, because glowing mushrooms, that's one of them.
That's a metabolic byproduct that's near and dear to my heart. And I know a lot of our viewers are probably excited to learn more about fungal bioluminescence. That'll be coming up at 1 o'clock. Couple more questions. Thanks for sticking with us, James. You've got a lot of people out here that want to know more information. Tanner asks, what does an average day in the lab look like for you surrounding this research?
Sure. Thanks, Tanner, for the question. A lot of the lab work that I do is maintaining my cultures. So the research is really seasonal based. So when it's raining, it's really great for mushrooms. And when it's dry, it's really not great. So I can't go out in the field in the summer looking for fungi. So I have to keep what I find alive. And so it's making more media. It's transferring fungi from media that they've eaten all of it to new dishes. So they have new food. Reading a lot of papers on wood decay fungi, and then doing those growth and those degradation experiments. So a lot of time just spent thinking about, the logistics. How am I going to make these wood blocks a particular size and keep it consistent? How am I going to put them into my mycelial mat? But yeah, it's a lot of culture maintenance. And then the degradation experiments themselves, they don't take very long. Yeah, thanks for the question.
The glorious life of a lab-based academic. It's nice. I think it's nice for our viewers to hear about both sides. Often people sort of glorify the fieldwork aspect of science and maybe sometimes forget that actually a lot of your time is spent sitting around reading papers.
Yep, that's a lot of it, most of it.
Which is fun too. Another question, Jan asks, how quickly would the mushrooms need to decay the wood in order for this idea to compete with using herbicides?
That's a good question. I don't know. It could be multiple years, right? But I don't think it would matter if it takes multiple years for it to completely degrade or, mostly degrade this stump. If it's preventing the resprouts while it's decaying it's okay if it takes 2 years or 5 years or 10 years to completely degrade this stump, because the goal of the herbicide is to prevent the resprouts. So if while it's actively decaying, if it's preventing resprouts, that works too.
Right. It just needs to be fast enough to stop them from growing back. Well, I think that wraps up our question and answer session, unless anyone has any last minute questions. James, this is really cool stuff. Again, I had no idea you were doing this research, and it's very interesting because I know the public would love to see an alternative for Garlon and herbicidal treatments. And how cool would it be if we could just use native wood rotting fungi to do that, to replace them? That's pretty neat.
I think it would be really cool. And so thanks again for having me. I really appreciate the opportunity. I've never been at the Fungus Fair before, so this has been really cool. I look forward to the rest of the presentations today.
Yeah, well, thanks for joining us. Hopefully next year you'll be able to attend in person and get to poke the tables of rotting mushrooms with everyone else.
I hope so.
It sounds like, folks, you can find James either working in Marin County Parks or doing his research right here in East Bay Regional Parks. So keep an eye out for him and say hi if you see him out there on the trail. James, thanks again so much for joining us today.
Thank you. Thanks, Trent.
All right, folks, we're going to take a quick break. And when we come back, we've got a little bit of time before our next presentation, Morgan Evans with Mushroom Cooking. So I'm going to give you a quick review of how I like to document fungi with my camera. Stay tuned. Welcome back, everyone, to the 2022 Tilden Fungus Fair. I'm your host, Naturalist Trent with East Bay Regional Parks. The slideshow you were just watching during our breaks from our good friends over at Coyote Brush Studios. They create handmade, sustainable, educational goods celebrating California's unique biodiversity and natural history. Artist Tina Curiel's original watercolor illustrations of California's native species are featured on a wide range of items, including stickers, greeting cards, prints, snack bags, magnets, and more. Coyote Brush Studios aims to inspire a love of loyalty by bringing the knowledge and imagery of California's native environments back into our homes and everyday experiences. And here's the best part. Coyote Brush is so stoked to be a virtual vendor for the Fungus Fair this year that they're offering our viewers 15% off anything in their store. Just use the code FungusFair, all one word, all caps, when you check out. I'll put that in the comments. Head on over to coyotebrushstudios.com and show our local artists some love. As promised, we've got a little bit of time before our next guest, and I thought I'd take that time to show you how I document the fungi I find in California and beyond. I'll preface this by saying I have no formal training in mycology. I studied botany and dendrology for my undergraduate degree, and I have learned our local fungi over the years using three tools: guidebooks, a camera, and iNaturalist. And today I'm going to implore all of you to obsessively photograph mushrooms just like I have over the years. So maybe you're asking yourself, well, why not just take everything home and identify it there? I mean, to collect the mushrooms and take them back to where your guidebooks are in the comfort of your kitchen or living room. Why bother crawling around in the rain and mud to get mediocre mushroom photos? Well, there are many times when you will want to collect everything, but maybe you can't. Maybe you're in an East Bay regional park where no mushroom collecting is allowed, or a California state park, or a national park, most of which don't allow collecting of any types of materials, fungi, plant, animals, etc. Right here in our own East Bay Regional Parks, in Ordinance 38, which is our big rule book, it says right there in Section 804, under plants, that no one can collect mushrooms. And I get a little bit of a kick out of this, and I'm gonna poke fun at my wonderful employer for not updating their terminology in this book, because, as you probably know, mushrooms aren't plants. And this fellow here, that's Dr. Robert Whitaker, actually published this information way back in 1962 when he dedicated an entirely separate kingdom to the fungi based on differences between common ancestors and different trophic positions, plants being producers and fungi being decomposers. But I digress. We're talking about documenting mushrooms here. So this is a perfect how not to photograph a mushroom photograph. It's from the top down, it's blurry, and there's zero context. This could be an apple or a dog chew toy for all I know. This is actually my mother's photo, so I can criticize it a little bit. Mom, if you're watching today, sorry. It's a perfect photo for this purpose. Don't take your photos like this. That makes it really hard to identify later. Um, Here's a little bit of a better angle, right? This person is capturing the top of the mushroom, but also the reproductive surface, the hymenopore, and the stem or stipe. In a perfect world, if you are trying to document mushrooms with a photo to identify later, you want 3 photographs. You want a habitat shot showing where the mushrooms are growing, because that is going to give you a lot of clues as to what the species is. Mushrooms are specific in their substrates and some of them in the plants that they associate with. So when you take a habitat shot like this, a nice wide shot, we can see that there are what looks like tan oak leaves on the forest floor there and some blackberries nearby. And that gives us a lot of clues. Then you're going to want to take a photo of the cap. And this is a cap detail shot. In this photo, we can see that this cap is white and has some prominent little triangular things. Maybe you could call them warts on the top. And that's an identifying feature too. And finally, you're going to want to take a shot that includes details of the stem and gills or fertile surface. Your mushroom may not have gills, it may have pores, it may have nothing at all, but it's going to have a fertile surface from which it produces spores. And you need to document that with your photo. So this is perfect, best case scenario here. 3 photos, cap detail, habitat, stem and gills detail. I did miss one thing from this photo montage, and I'll talk about that in a little bit, and that's a scale. Without scale, we don't really know how big this mushroom is, and it was quite large. It was about 11 inches tall. This is Amanita magnivaricata, which is a fairly charismatic and easy to identify mushroom once you get to know it. So here's an example of a combination shot. This shot was trying to capture all of those details in one photograph. So we've got the cap, we've got the stem, we've got the hymenophore, the fertile surface. In this case, it's a layer of pores. We've kind of cropped out something. We're kind of missing a little bit of the habitat shot. I guess the habitat for this one is moss, but if I'd pulled back a little bit with this photograph, I might have gotten some leaves from nearby trees or some plants that could have given us clues. Here's the thing not to do. Don't use your phone's built-in flash because it usually looks really bad. This is the kind of photo you'll get when you use the flash on a mobile device, overexposed, blown out highlights, kind of impossible to recover highlights and contrast. This is Swillus umbonatus up in the Jackson demonstration forest. And again, luckily, it's relatively easy to identify from a photograph because it has this characteristic glutinous veil that you can see there that's still attached to the edge of the cap. The veil or partial veil is what covers the fertile surface as the mushroom is kind of coming up out of the soil. It keeps those spores in until they're ready to be dispersed. So instead of using your phone's built-in flash, there are other simple tricks you can use. I took this photo just using a sheet of white paper to reflect sunlight onto the bottom of the mushroom. This is called a reflected light shot, and it often will turn out a lot better. Now, you may need a tripod or something to hold your phone still or your camera still enough, or you can kind of juggle 2 hands, have your camera in one hand and have your reflector in the other. It's not impossible, and if you try it, you'll probably get better results than your phone's built-in flash. This is Sarcodon imbricatus up in Alaska. It's a tooth fungus, very charismatic, also very large. Sometimes you may want to add a few more details to your documentation shots. This is an all-in-one photo here, and I've got a okay view of the cap. I've got the fertile surface, the pore layer. I've got the stem. I've got the habitat. And those are coast live oak leaves in sandy soil. But there's something about this species that would help me learn more and have an almost instant identification. And that's taking a physical cross section of the mushroom. So this is Swillellus amygdalinus. And when it is bruised or damaged in any way, it very rapidly oxidizes and stains blue. And so that's a telltale characteristic of this mushroom, something that can help you get to a quick identification. So again, sometimes depending on the species you're trying to document, you may want to think outside of the box. If you aren't taking your collection home, there's something else you may want to add. As I mentioned earlier, this photo shows a poured hymenophore, dried cracking cap, blue staining, but I'm missing something, and that's a scale. So when we throw the scale in, then that adds a new element that can help us identify our photos after the fact. So this was a very large mushroom, and this is, I'm completely blanking on what species this is. This is some type of bolete. I think it's a Xerocamella species that I photographed up in the Mendocino National Forest. So why do all this stuff, right? Well, again, it's to document your finds so that you don't have to bring it all home. Or even if you are collecting and bringing your mushrooms home, having a photograph, especially a geotagged photograph, can help you so much in both identifying what you're looking at and saving a record for later, particularly if you upload it to iNaturalist. So that is the main reason that I like to take photos of mushrooms, is so that I can upload them to iNaturalist and then have documentation of everything that I have seen right down to the location. So this is a map of just my mushroom observations here in Tilden Nature Area. And this has helped me a lot over the years in both learning our local mushroom species and having a reference that I can go back to. So I can remember when things were fruiting and where they were fruiting and what they looked like. Now, we touched on this a little bit yesterday, if any of you are watching and also watched yesterday, and there's a certain best practices or an ethic that comes along with sharing information on the internet. Let's call it social media best practices. iNaturalist is a public platform, and so when you upload any observations to iNaturalist, they become public information, public knowledge. Anyone can go onto iNaturalist with or without an account and view where you saw your things. So here is a very large Cantharellus californicus, the California golden chanterelle that I collected, and this is something that I am not going to upload to iNaturalist for several reasons. One, because I'm going to be protective of any legal chanterelle spots that I find, because they're few and far between, and I don't want to cue commercial mushroom collectors into them. And I guess the second reason is I don't want to cue commercial mushroom collectors into them. I like it if people go out and find their own mushrooms, particularly commercial collectors. And so I personally am not going to upload choice edible mushroom observations to a public platform. However, if I really wanted to share the location of this chanterelle, or at least the fact that I observed it and the time and date, I have an option, and that is I can obscure the coordinates of it. So that's an option for any time you upload anything to iNaturalist, you have the choice to obscure the coordinates. And what that does is it puts a really large error radius on your observation so that no one can pinpoint exactly where you saw it. And someone will know that, oh, hey, Naturalist Trent found a chanterelle down in the Santa Cruz Mountains, but they won't be able to see exactly where, and therefore won't be able to go directly to that spot and, pick every chanterelle that grows there for the next 5 years. So I use iNaturalist with some caveats, and it's going to be up to all of you if you choose to use this platform to determine your own best practices. So I know we talked a lot about this yesterday, but I'm going to plug iNaturalist again. Folks, it's extremely easy to use. It has both a desktop and a mobile interface. The mobile interface is particularly good because it uploads directly from your phone. It cuts out a lot of the user time, and it reads the geotags and metadata tags and applies them. So your phone, if it's set to record geolocation, that tag will automatically go to iNaturalist. So you don't really have to do anything except push to upload. And of course, I really appreciate several other parts of iNaturalist. One is the sort of social media aspect, and Christian talked a lot about this yesterday, but there's a really big community of people that use iNaturalist. And that community is made up of real people that are passionate about the natural world and also passionate about helping you identify things and sharing your stoke. Here's just an example of a California pink glowworm that I observed. And, we had this robust mini page conversation about this because people were really excited for this observation to show up because it's a cool bug. And one of my favorite features of iNaturalist, and this is relatively new in the grand scheme of things, it's been out for a few years now, is not HAL 9000, is the photo recognition. So iNaturalist and its sort of simplified partner app Seek both have the ability to take a photo that you upload and read it and compare it against the massive database of photos that are stored on the iNatural servers, and then give you a suggestion for the identification. And I have to say, the suggestions are pretty good. You should never use a photo recognition suggestion alone to determine edibility of a mushroom or a plant. But if you are casually observing something, these suggestions are a great place to start. And it can cut out a lot of the legwork on your end of going through a taxonomic key or a binomial key to get to a place in your guidebook. In this example, I took a photo of, actually, this is a photo of some Amanita Calyptroderma mushrooms, which I was just doing as a demonstration. And there the app suggested, hey, we think this is an Amanita. And the first suggestion was, in fact, Amanita Calyptroderma. Even if I didn't trust it, I could say, okay, well, Amanita, I'll go to that chapter in my mushroom guidebook and I'll figure it out for myself from there. So this photo recognition technology is really fantastic, and it works quite well in the Bay Area, where there are lots and lots of identifications to pull from. So lots of different examples here of the software getting it right, and some misses here. Sometimes it'll say we're not confident enough to make a suggestion or recommendation, but here are our top 10. So it might be one of these things, and in this case, it was the third suggestion down. Now, one drawback to citizen science platforms, and again, apologies for repeating this information from yesterday, is that the iNaturalist doesn't observe the real world occurrence of fungi or plants or really anything, because it's not based on a design study. And so all these observations are heavily user biased. For example, this map, that's the Tilden nature area. And you can see that there are lots and lots and lots of observations within the Tilden nature area, that circle. But once you go outside of that, there are quite a few less. And that's because that's about the distance I like to walk from my office on a general day. So that's my observer bias built in, and it's showing on this map of observations. Despite this, iNaturalist is still a really, really useful tool for really anyone who's interested in learning more about the natural world. Professional park naturalists, park rangers, resource managers, land managers, scientists, and more. So it's a fantastic tool. And when it gets used enough, we start to be able to notice landscape level trends. So this is an updated screenshot of a project that I started about 8 or 9 years ago, Fungi of the California Floristic Province. This project just aggregates all mushroom observations that have been seen within the California Floristic Province, which is a boundary that was kind of written up a long time ago by folks who studied plants. But as you can see, there are 500,000 observations of fungi now in the California Floristic Province, and almost 3,200 species. That's a lot. Before iNaturalist existed, we had no idea of knowing this because mushrooms just weren't very well documented. So again, I implore you, take pictures of your mushrooms when you're out and about. Share them on iNaturalist if you think it is appropriate to do so, and become a part of the community science community and contribute something to the greater scientific understanding of our world. It'll make you feel good. All right. So a couple of comments here. Let's see, how quickly that was from the last one. Redisclosing locations. Also know that if you have geolocation on your camera, your photos have that location attached. So anywhere you post them will disclose that info. Krista, that's partially true. A lot of social media sites will scrub that location from your photo if you upload it. iNaturalist won't, because iNaturalist is designed to use the geolocation from your photo in order to put a place on it. And you can change the settings in your phone, or you should be able to, depending on your phone, to actually scrub that geolocation data from that photo before it leaves your phone. So, for example, if you text it to someone else, you should be able to scrub that geolocation information off of your photo before you share it with the greater world. But good point. If you are taking geolocated photos, just be a little careful of where you send that and share your information. All right. And with that, folks, we're going to take another quick break, and we will be back in just under 5 minutes with mushroom cooking with Morgan Evans. Stick around. All right, everyone, welcome back to the 2022 Tilden Fungus Fair. I'm your host, Trent, broadcasting from Tilden Nature Area. That slideshow you just saw was from our friends at Coyote Brush Studios, who are offering 15% off anything in their store to our viewers today. Head on over to coyotebrushstudios.com and use the code FungusFair when you check out. I just dropped that link in the chat. Folks, I've known my next guest since I started working at the Tilden Nature Area back in 2012. She began her career as a docent for the Tilden Nature Area. She then joined our staff of interpretive student aides while working on her Ranger Naturalist certificate, which is when I met her. She also worked as a docent at the Regional Parks Botanic Garden, a naturalist aide at Big Break, and a recreation leader before becoming a full-time recreation coordinator based out of our Tidewater Boating Center. Morgan Evans also holds an associate's degree in culinary arts, and she loves to merge her two interests, the outdoors and food. She's also a certified interpretive guide and loves backpacking, botanizing, and foraging for wild edibles. Morgan, thanks so much for joining us.
Thanks for having me, and thanks for the introduction. So again, my name is Morgan, and I've been doing mushroom-themed programs here in the nature area as far back as 2010. And I'm very excited to be back here today sharing one of my favorite subjects, mushrooms. Specifically, my favorite choice edible, the black chanterelle. The Latin name for this mushroom is Craterellus cornucopoides, and that translates to Craterellus means small cup, and cornucopoides translates to horn of plenty. This mushroom has a lot of different common names, including some that can be somewhat daunting. The mushroom of the trumpet of death, the black horn of plenty. In any case, this mushroom is one of my favorite choice edibles. If you've ever been on a mushroom hike with any of the staff here, people invariably will be asking, is it edible? And so when we talk about edibility with this mushroom, we're going to call it choice. Just because something's edible doesn't mean you're going to want to eat it. So it ranks up there with the porcinis, with the morels, with the boletes, and the true chanterelle. So this mushroom, again, is a Craterellus. It is not a true chanterelle. The true chanterelles are Cantharellus, and that translates to drinking vessel. So very similar. It's in reference to the shape of this mushroom, but again, not a true chanterelle, but a fine mushroom in its own right. So today, I'm going to go over some fun facts about the black chanterelle. Then I'm going to describe all the ingredients that we're going to be using here today to create our dish. Actually, 3 dishes. We're going to have one recipe that's going to have 3 different preparations. So you can use this recipe in different functions. And then I'm also going to go over some of the equipment that we're going to use, and then we'll delve into the recipe itself. So again, this mushroom is my favorite choice edible. It took me a long time to be able to recognize it in the wild. It has a habit of kind of hiding itself in the forest floor. It tends to grow on the ground, on stream banks, creek sides, road cuts, along trail systems. But you have to really have an eye attuned for its form. So people say that it looks like a black petunia, or that there are black holes in the ground. Once you've attuned your eye to what it looks like, you'll see them everywhere. So where they do grow. So they do grow along the coast. They are, if you do find one, you'll often find a lot of them. And the proper way to harvest them is to not cut them straight on the ground, because that way you'll get some dirt and grit in here. One of the reasons I love this mushroom is that it tends not to have a lot of dirt. You can see this mushroom is actually pretty clean, right? So whereas regular chanterelles, the golden chanterelles, tend to have a lot of dirt. You have to spend countless hours cleaning them once you've brought them home from places where it is legal to forage. But this mushroom tends to be ready to go. Sometimes you'll find some insects in there. Sometimes you'll find some pine needles or something, but they're very easy to clean. Another fun fact about this mushroom is that the underside of this, which is actually the hymenophore. Trent mentioned the sporing portion of the mushroom. So as you notice, this mushroom does not have a cap that's separate from its stem or its stipe, but this whole outer surface is where it's going to release its spores. So we call that the fertile region. That's the hymenophore. This portion of the mushroom, it actually reflects light. So if you were out at night and you were using a flashlight, you would actually see this thing reflecting back at you. So fun fact, you can actually forage for this mushroom in the dark. Some other facts about this mushroom, it associates with tan bark oak and mixed conifers, madrone, those type of forests. So it's where I like to go looking for this mushroom is up the coast on the Sonoma-Mendocino border. If you are on a social media platform, this year it seemed like everyone was finding this mushroom. So I decided that because everyone's harvesting it, who are finding it, that I would showcase this mushroom, my favorite choice edible, in today's preparation. So enough said about the mushroom itself. Our recipe today is going to be a meatball. And so I'm making a chicken meatball with black chanterelles. You can either use fresh black chanterelles. Actually bought these at Monterey Market. I did not have any time to go foraging myself. Sad for me. Or you can use dried black chanterelles as well. So the dried form actually has a more concentrated flavor. They dry very easily. They actually keep very well in the fridge. These mushrooms here, they've been in my fridge for probably about 10 days now, and they really haven't suffered very much. You can keep these in the fridge for up to 2 to 3 weeks. Another reason that they're very convenient mushroom. They dry very easily. You don't even need to clean them before you dry them because you can actually just kind of sift through it and have the dirt fall to the bottom. In fact, if you use the dried chanterelle in this recipe, I would just rehydrate it and then squeeze out the moisture and then chop it up and you're good to go. Today, however, I'm going to use the fresh ones just to kind of contrast them. But again, if you're looking for something with even more kind of intense mushroom flavor, you might want to choose the black chanterelle. If you have a copy of Christian Schwarz's book, The California Mushrooms of the Redwood Coast, he mentions that some people who harvest this mushroom will just dehydrate the whole lot, which is what I tend to do because, again, they kind of grow in profusion where they do grow. So often you don't have a chance to eat all of them. So drying them is a perfectly reasonable way to go if you find that you have a lot of these on your hand. So again, we have our black chanterelle or horn of plenty. We're going to be using fresh tarragon, one of my favorite herbs. Panko, which is a Japanese breadcrumb. It's a wheat-based product. It's one of the only products that I buy that contains palm oil, unfortunately, but you can use any sort of breadcrumb that you want. If you have some homemade breadcrumbs, or you can buy breadcrumbs in the store. I try to steer away from the ones that have other seasonings in them because I want to be able to control the flavors of my dish. We're going to have some pistachios. We're going to chop these up. So these are roasted and salted. Often when you're making a meatball, it's kind of hard to get a lot of flavor into the meatball, especially the salt. So I'm going with ones that are already roasted and salted. Again, the mushrooms, you're going to need some fresh eggs. Here we are at the little farm. I didn't have a chance to go shake down a chicken this morning, so this is from the store, or nice organic eggs. We've got some grated Parmesan, Romano blend, fresh garlic, of course, always, and some porcini salt. Now, if you've never made your own flavored salts, we're going to do it really quickly just to demonstrate how easy this is. You can make it with any sort of dried mushroom. You could even make black chanterelle salt. In fact, I think that's what we'll do. We're going to make some black chanterelle salt. So it's going to require a clean coffee grinder. So please don't use the one that you grind your coffee in because it has all those oils. It's going to be really hard to keep that clean. I have one that is designated solely for different types of herbs and whatnot. So we're going to just take some of these black chanterelles, trumpets of death that I dehydrated myself. We're going to put them in this coffee grinder with some kosher salt. Again, this is coarse kosher salt. So I'm actually going to grind it together. Otherwise, you would just grind the mushroom separately and then add it to the salt itself. So it's going to go ahead and grind this up. See how it looks. It's a little chunky, but that's all right. You can grind it a few more times. Voila. We've got some black chanterelle seasoning for our meatball. If you want to clean out your coffee grinder in between different spices that you're grinding, just use a fresh piece of bread, process it, and then dump it out. It's super simple. So again, you can do this with any sort of dried mushroom, dried shiitakes. Often dried mushrooms have a much more concentrated flavor, as you would imagine. I'm good friends with James Yu, who owns Great China, one of our fabulous Chinese restaurants here in Berkeley. And in their dishes, they strictly use dried mushrooms that are rehydrated because it has that super rich umami taste. So, okay, we've got our salt here. We're going to have some freshly ground nutmeg in our recipe, and of course the ground chicken. Now the chicken itself, it's going to be hard for you to find a butcher who will grind up the dark meat as well as the white meat. If you go to your local butcher, they're generally going to have just ground up white meat. But for a recipe like this, you need a little bit more fat. And so I had to go to this shop called Local Butcher and have them grind up the chicken meat for me specifically. That's about it for our ingredients, besides, of course, some extra virgin olive oil and some freshly ground pepper. So now I'm going to go over some tools. Now, you see a lot of tools here, and there's a lot of tools. If you go to Sur La Table, you're going to see like 5,000 different types of tools. When I travel, the main things I bring with me are my own personal sharp chef's knife, a really nice paring knife, and a garlic press. Everything else is pretty, you can kind of make it up, but these are the three things that I don't go anywhere without. I happen to like these type of knives that are a high carbon steel. They are very easy to sharpen if you sharpen them yourself, but they will rust because they don't have a lot of stainless steel in them. They're more of a high carbon steel. So this is the type of knife that I prefer, but as you like. Some other things we have are some fresh ground pepper, some measuring implements, and later on for one of our preparations, we're going to need a pastry brush. So the first thing that we're going to do is we're going to cook our mushrooms because we want to get them cooked and then set aside to cool before we mix them in with our chicken mixture. So I'm going to turn this up a little bit. I've already cleaned these mushrooms here. I bought these at Monterey Market. They have an amazing mushroom selection. If you're looking for anything off the beaten path, that's the place where I would recommend going to buy mushrooms if you don't have the savvy to go looking for them yourself. And again, no collecting here in the East Bay Regional Parks. There are places that do allow foraging. Ones that come to the top of my head are Salt Point, which is up the coast, again, Sonoma, Mendocino area, and Point Reyes. But of course, this year, with the pandemic, everyone has been out and about trying new things and creating new hobbies for themselves. And so I think these places can tend to be quite overrun. Nice thing with the black chanterelle, however, is that this mushroom will have multiple fruitings throughout the season. So the season for this mushroom is midwinter through spring. I found these mushrooms as late as Mother's Day up the coast. So if you go up to the coast and you find that everyone and their brother's been up there collecting mushrooms, don't despair. You just might need to go at a different time of year and see if you can have your luck. Often you'll find where people have cut the mushroom and then there'll be little baby ones growing up alongside of it. Again, mixed conifers, hardwoods, tan bark, oak, and madrone manzanita type of mixed woodlands. All right. These are going to shrink as we cook them. I'm going to cut them up just a little bit more. I'm doing this ahead of time because otherwise, once they're cooked, it's going to be kind of messy to cut them up. This is quite a lot, but that's all right. We want a lot of mushroom flavor in our meatball. So I think this is hot enough now. Going to use some extra virgin olive oil, Bioluminescence, to cook our mushrooms. Hopefully everyone can see that. Turn it up just a little bit. It's pretty hot now. These mushrooms do not require lengthy cooking. They're very thin-skinned or thin-walled, and so they're very different from an actual chanterelle, which is very thick and kind of meaty, kind of fleshy. So again, just needs a few minutes in the pan. I'm going to add some fresh ground black pepper. It's always nice to have fresh spices. And then some of our black chanterelle salt. We're going to go big on the chanterelle flavor. And again, if you didn't want, or if you didn't have the fresh mushrooms, if you had a supply of the dried ones, you could just very easily rehydrate them in just a small amount of hot boiling water, let them sit 5 or 10 minutes, squeeze out the liquid, and I would just put them straight in the recipe. That way you wouldn't even really need to cook them because these really don't require a lengthy cooking process. They're not like a matsutake that's really firm and kind of chewy, has a lot more chitin in it, is my understanding, than something like this that just cooks really right. And you notice that it's turning darker in color, so I think we're getting close to calling this done. They will cook as well in the meatball and in the ravioli. So I'll go ahead and turn this down. I'm going to put these aside to cool. You can smell them. Has a very earthy aroma. Another fun fact about this mushroom, I pulled out my supply of dried chanterelles. Yesterday I was making different versions of this before today. I made 2 different versions of this before today. And my cat, his name is Tristan, went crazy over these black chanterelles. They were like his favorite snack food on the planet. So I'm going to nickname him Umami Cat. He tends to like the finer things in life, goat cheese. I had no idea he has such a fondness for black chanterelles, but apparently he does. Okay, so I'm going to set this aside and let that cool. And then we're going to work on the rest of the filling. So I've already portioned everything out. I do believe there is a copy of this recipe that will be available to you all. I'm really not good at measuring, but I did it this time just to be on the up and up. So we're going to start in our bowl with the panko or the breadcrumbs. We're going to add a little bit of dairy product to this. You can use regular milk. You can use heavy cream. I'm using half and half. I'm going halvesies here. You're going to add this to your breadcrumbs just to rehydrate them and get them kind of moistened in advance. Stir that around. These are very crunchy breadcrumbs. Then we're going to add our grated Parmesan Romano. I like a lot of cheese in my meatballs. I learned that from A16, putting cheese in your meatballs. We also need to chop up our pistachios. You're going to want to chop these up relatively small, just because the meatball. For some reason, the chicken meatballs tend to be much softer in texture than one made with beef and pork. And so we don't want big chunks of the pistachio in there to kind of break apart the meatball. We want them fairly small. If you had a Cuisinart, you could very easily do this in much less time. Being careful not to cut yourself. And again, roasted and salted. We want that good salty goodness in our meatballs. All right, call that done. I'm going to put that into the breadcrumb mixture. And you could use any sort of nuts that you like. Hazelnuts might go really well with this as well. It's just to your liking. We've got our fresh tarragon. Chop that up. This is one of my favorite herbs of all time. Has a kind of licoricey kind of smell. It goes really well with seafood, any sort of type of fish, and chicken as well. Toss that in. Garlic press. This is the world's best garlic press. They stopped making it. The brand is Xylus. So if you ever go to a Goodwill or a consignment shop and you see one of these, grab it because they no longer make it. It's indestructible and it is absolutely positively the best. I like a lot of garlic. Much to my partner's chagrin. He's a super taster and it always tastes too garlicky to him, but not to me. And yeah, if you wanted to kind of mellow out the garlic flavor, you could also alternatively cook it with the mushrooms. But I'm just going to be lazy and throw it in here like that. Mix it up a little bit more. And I'm going to add some more black pepper to this. Now, before you go ahead and form all your meatballs, you'll have a chance to make a sample one and cook it in your skillet or your pan, just so you tell that you have the seasoning correctly. Again, sometimes it's really hard to get the proper amount of salt in something like this, and then you form it all, and then it's too late. Other secret ingredient, some nutmeg. This thing is also a handy dandy little device. I don't go in for a whole lot of gadgets, but this one, the nutmeg grinder, it's very keen. It's always nice to have freshly ground nutmeg. All right. So, the eggs. We want to beat the eggs a little bit before we put them in. You want to mix everything together up to the point where you add the chicken, because you just want to very gently incorporate everything into the ground chicken. If you overmix it, it's going to tend to get tough. Same would go for beef or pork meatballs, but especially with the chicken meatballs, and especially if you didn't have ground dark meat. So just enough to get it mixed up. We're going to leave a little bit in there for our ravioli. Set that aside. Okay, and what's missing? The mushrooms. Hopefully they've cooled off by now. Yep, cool enough. Gonna add those in. So this mushroom has another common name. and that is the poor man's truffle. And I thought, oh, well, I guess that must mean that it tastes somewhat like truffles. Well, it has a very ethereal, very earthy aroma to it, but not something that you would compare to a truffle, which is pretty much hits you on the head, right? Drives truffle pigs wild. This mushroom, they called it the poor man's truffle because people would shave these little bits and it would look like a shaved black truffle. So in this book here, one of my favorite cookbooks that talks about wild forage foods, I think last year's recipe came out of this book as well. She has a recipe for the poor man's truffle, which includes, of course, the trumpet of death. All right, so I've got this mixture pretty well incorporated. And now we're going to go ahead and add our chicken. So I asked the butcher to grind me up half white and half dark. You just want a little bit of fat in there in this recipe to keep it moist. This nice paper. But I had to call around to multiple places to find someone who would do it. I'm going to try to just fold this in and not use my hands. I can already tell this is probably going to need more salt. But we're going to do a little test run and make sure that it has enough flavor in there. Otherwise, what's the point? Nobody wants a bland meatball. All right, maybe just a touch more salt. You can tell I don't have high blood pressure. I'm pretty liberal with the salt, but of course you could not put as much salt in there, depending on your dietary needs. All right, that's looking pretty good. I think we'll do a little test patty to see how we did on our seasoning. It's good and hot. And again, these are going to be really soft. Much softer. Of course, there's a lot of stuff in here, right? To kind of keep it from holding together. Back in 1 second. Rinse my hands. Turn this up. So the nutritional breakdown of this mushroom, just while this is cooking, I will read off some stats to you. So 100 grams of the dried black chanterelle has 70 grams of protein, 14 grams of carbohydrates, 4 grams of fat, and approximately 380 calories. It's also high in vitamin C. It has about 87 milligrams of vitamin C. So health food, as well as being delicious. It is also nutritious. If you've never had black chanterelles, I would really recommend going out and trying them, experimenting with them. There's lots of things that you can do with them besides just the recipe that I'm making here today. Some of my favorite ways to cook them are of course, in a risotto. I also like to make a ravioli filling with roasted butternut squash, sauteed leeks, garlic, thyme, and we'll show you the technique as to making the ravioli filling as soon as we're done with our meatballs. You could use this. This is actually a recipe from a cookbook I no longer own. I've adapted it, but it was a recipe for a breakfast sausage. So you could also just make this little sausage patty to go with your eggs. Again, black chanterelles, eggs, classic pairing. One of my most memorable meals I ever had in my life was at Oliveto Restaurant. They had these specialty dinners throughout the year. They had like a tomato one, a balsamic one, but they had one that they called the whole hog dinner. And it was like the best meal I've ever had. But they had a freshly ground red heirloom corn meal polenta, some sort of pork product that I'm not remembering right now, and the black chanterelles. And I think it was one of the best meals I've ever had. So this is looking nice and brown. I'm going to go ahead and give it a taste. Make sure I've got the seasoning correct. The garlic off my knife. It's going to be a little bit hot. Tastes pretty good. I am going to add a little bit more salt though. And just try to gently fold it in so as not to make this tough. Okay, so I think at the beginning I mentioned that this is one recipe that's going to have 3 preparations. Your first preparation you see here is simply to make it into a meatball. So we'll add a little bit more oil to this pan. We'll cook off a few of these meatballs. You can brown them in the pan and then finish them in a hot oven because they are so kind of soft. You don't want to manipulate them too much. You could also just make them straight into the pan, into the oven. I wish I had a live audience. You all could experience all the senses going on with these mushroom balls right now. So I'm just going to brown these. On a couple sides, and then we're going to put them in the oven. Being impatient here. So this is a very simple appetizer. Or again, a breakfast, excuse me, type of sausage. I'm going to put these on my pan. I'm going to pop these in the oven. And then we're going to talk about the second preparation to go along with this one recipe. All right. Another great appetizer is a stuffed mushroom cap. So we have these creminis. Going to take out the stems. So we have the empty filling, center for filling. You want to toss them in a little olive oil. Salt and pepper. Go with my newfangled black chanterelle salt. So we'll get these a little bit seasoned, moistened, ready to go. You could also do this as an entree. You could use a full-size cremini, which is basically a mature portobello. Portobello is a mature cremini. If you wanted to go entree size with these, if you're having a party and you want to impress your guests, what better way than a stuffed mushroom cap? Go ahead and set this aside a little bit. Just going to pick up some of this filling. Very simple. Stuff the caps. And these are going to go in a 400-degree oven for approximately 20 minutes. Approximate because I don't know if your oven is calibrated. Go ahead and put these on a separate tray. Excuse my back. There. Take the rest of this morsel. Give me strength. For our third and final preparation for this one recipe, it's really good. You can actually really taste the chanterelle. Is to make raviolis. So don't freak out. You don't have to make fresh pasta for this. It's a very simple way to make them. You can buy pot sticker wrappers. They come round, or there's different ones that come in a square shape. Either one works. Very quick and easy way to make some raviolis. Gonna go back to my egg wash. Here's the part where you need the pastry brush. I'm just gonna put the egg wash on one side of the wrappers, but I'm gonna cover it completely so you can see that. They put some coating on here that keeps them from sticking together, and that's fine. You can just leave that on there. It doesn't bother the final product. And here you're going to want to be cautious about the amount of filling that you put into these. If there's too much in there, they're just going to pop open. You need to have enough of the edge around here that you can secure, that keeps it together when it goes into the boiling water. So even that amount might be too much. Go a little bit smaller, but make sure you have enough of the black chanterelle in there. And again, if you wanted to go meatless, roasting a butternut squash, mashing it up, the sauteed leeks, goat cheese, thyme, and the black chanterelles makes a really lovely entree around Halloween. If you have like doing themed menus, it's a perfect thing to do. When I worked at La Lime's restaurant in Berkeley many moons ago, we always had black chanterelles on the menu around the holidays. So then I'm going to take my top wrapper, put that on my way, and I'm going to press it down. And what I'm trying to do is get all of the air out of the inside of this wrapper. Otherwise, it's going to float up and the air is going to try to escape and you might have a break. So once I feel like I've gotten the air out of there, I'm just going to press down pretty forcefully on the sides, on this edge to get it secured. Do that with all of them. Makes very quick and easy work if you wanted to have homemade raviolis, make your own filling. The stuff, the ones you buy in the store, you know, at Trader Joe's, they're okay. But if you want to make it yourself, this is an easy way to do it without having to make fresh pasta. I'm going to, whoops, press that edge down. And I'm going to let these sit. I'm actually not going to cook them today because I don't have boiling water here. But I would let them sit for a few minutes before I went ahead and boiled them. Salted, boiling water, 4 or 5 minutes. Voila. Maybe now we can check on our meatballs, see how they're doing. Just want to make sure that they're cooked through. While we're waiting on that, I wanted to share this botanical illustration that I found on the internet when I was looking for images of the horn of plenty, the trumpet of death, the black chanterelle. And I just thought this was really funny because the one description of this mushroom says unpalatable. So someone tried this mushroom and was like, no thanks. I happen to think it's one of my absolute favorites. Let's see how things are going. Take out these. These are sizzling. Mushrooms back in. So again, those are going to take about 20, 25 minutes. But here we're going to pretend like I cooked the raviolis, which I didn't. And these are going to be our 3 recipes, 3 preparations. We've got our meatball, our cocktail meatball. You can serve it with toothpicks. Garnish it however you'd like. On a bed of something, I don't know. Our stuffed mushroom cap, and then our ravioli. You can go fancy and drizzle this with a little bit of truffle oil. Who doesn't love truffle oil? It is can be overpowering, so if you wanted to just get the flavor of the black chanterelle and only that, I would suggest maybe not doing that. But I happen to love truffle oil myself. And some grated Parmesan. You can make it very simple. You can make a nice cream sauce, a very simple beurre blanc, where you would saute some shallots and garlic, reduce that down with some vermouth, and hit it with cream at the end, and just kind of let that reduce a bit. Add whatever herbs you like, whether it be thyme or more tarragon. It is entirely up to you. So here are our 3 preparations. They look fabulous. I wish I had an audience here to try them out with me. But now you're probably thinking, okay, I have these amazing appetizers. What am I going to drink with them? So while I did my 2 trial runs at home, we tried them with this particular Sancerre 2019, and it has a bit of earthiness. So it's very hard to find earthy qualities in wines. Often they're very fruit forward, but and especially in a white wine. In red wines, you're going to look for something like a light red Burgundy, like a Pinot Noir, or even a white Burgundy, a Chardonnay. So as the Pinot Noir ages, often it starts off with your fruit flavors and some mineral flavors, but as the fruit kind of dies down, when it's more aged, that's when the earthy qualities come back in. So I would pair something earthy to go with this dish, just because we've got these amazing mushrooms here. See how this Sancerre is holding up. With, what should I try? Which one should I try? Eeny, meeny, miny, mo. I'm going to go for the meatball, just because it's the freshest one here, even though I already had it. Mm. Delicious. Excellent, excellent pairing, I have to say. But again, if you're not a white wine drinker, if you prefer a red, you want to go with an aged Pinot Noir. If you're on a budget, you could imagine having this with Schiava, which is something from northern Italy. It's a lighter bodied red, but will also have some earthiness that will hold up to the earthiness of the mushroom. And with that, I think I've talked about everything I wanted to talk about. See if anyone has any questions. Stump the culinary professional. It's your opportunity. I'm ready. Maybe you have a question. Or maybe we're not ready for questions yet. I'll go back to eating my mushroom. Meatball. A full-size entree with this mushroom. Go right for the portobello.
Folks, we're back. Thanks for bearing with us there. I was really hoping I would get to use that technical difficulties slide I made. If anyone has questions for Morgan, go ahead and put them in the comments section and I will read them out to her. Morgan, that looks amazing.
It's pretty good, I have to say.
I
do say so myself.
I'm a little embarrassed to tell our audience here that I actually get to go and taste this as soon as the show's over because Morgan is presenting and cooking from our auditorium right here in the nature area. So lucky me. I know. A few questions for you, Morgan. Mark wants to know, can you substitute oil or butter to make up for fat if using only white meat ground chicken?
You know, I asked the local butcher fellow who was ringing me up, how would I incorporate more fat into this dish? So if you were just using white meat, he said you could incorporate some fat, like if you had some duck fat lying around or, gosh, not really sure. But if you were to use heavy cream mixed in with the breadcrumbs, you know, the nuts are pretty fatty. When I made it the first time with just the white meat, they were kind of dry. And what I ended up doing was I made that beurre blanc that I was telling you about, and I cooked the meatballs in the sauce, and that helped to kind of tenderize them. But you could always try to add some duck fat. You can go with the heavy cream route, but I would certainly highly suggest getting ground dark meat. You can't find a butcher who'll do it. Local butcher, which is downtown Berkeley, it's Cedar and Shattuck, did it for me, but it was not cheap.
Well, probably worth it to make such a delicious dish.
Mm.
Sarah asks, do you know if rehydrated mushrooms can be used in the same quantity as fresh mushrooms would be used? Is there a difference in concentration of flavor and or texture?
Oh, the dried ones are going to have more flavor. So Trent and I, we disagree on this point. When he finds boletes or porcinis or ceps, he likes to cook them and eat them fresh. I take them, I immediately dehydrate them because I want that concentrated flavor, right? So dried mushrooms just in general are going to have more flavor. It's a perfect way to make that porcini cream sauce. Often you find a mushroom in the wild, they've got bugs. Another reason I love the black chanterelle. There's so many great things to say about this mushroom. No maggots, right? Super straightforward. But as far as quantity wise, you know, the recipe calls for 6 ounces of mushrooms. You would just have to eyeball that, whether it's weight or volume, right? So And, recipes, I have a very hard time following them. I like to eyeball things. And so I want to make sure there's enough mushroom flavor in this meatball. I don't want it to just have a few pieces in there. You're not going to taste it because the whole point is to showcase the mushroom. So just add some, do a trial taste run, and then add more if you want. But once you hydrate it, you can imagine that it's just kind of like the fresh one.
Okay, interesting. Well, yeah, we'll have to agree to disagree on that. And to be fair to our viewers out there, I also like dehydrating my porcinis and other boletes. But yeah, I just kind of sometimes you just got to have that big fresh slab of mushroom that you fry up right then.
Yeah, grilled porcini, a fresh one.
They're pretty
If you
good.
get them before the maggots do, for sure, right?
I don't mind the maggots. I think it's part of the whole wild mushroom eating process. But I'll stop that line of thought before I ruin everyone's appetite from your delicious meal you've just prepared. Mayumi asks, for the black trumpet salt, can you use it as a finishing salt since black trumpet is not cooked versus porcini does not need to be cooked?
Yeah, of course you can. Sure. Yeah.
All right.
You could use it like any other salt.
Krista has a similar question about using something like morels in a morel salt or meat rub. Would it be okay as a finishing salt since morels must be cooked?
Yeah, I don't know if you could use a dried morel. I guess you probably could, because I imagine that those chemicals that are like rocket fuel aren't still in that mushroom when it's dried, as long as you're not inhaling or whatever. I think you probably could.
I think a lot of folks, I think a lot of folks dehydrate their fresh morels and then use them after that. I don't know. That might be something for our viewers to follow up on. At what temperature does
try it and then let me know how it goes. I'm intrigued now, right?
I was thinking maybe they could research it before they tried it for themselves. But at what temperature does the MMH or whatever that compound is called in morels evaporate off or volatilize would be the question. And will your dehydrator do that, thereby making morels safe to eat?
Yeah, it might make you sick, right? Yeah, absolutely. Do some research.
Don't want to end up like that chef up in Seattle or wherever that was that fed fresh morels to a huge group.
Oh,
another different story. Right. And Sarah asks, I've heard a lot about how mushrooms are a great way to get the umami flavor in a dish. Is there a range of this in mushrooms? For example, are cremini more meaty or umami-ish than an oyster mushroom?
Oh, yeah. With the choice edibles, I would say in my mind, the porcini is probably like the king of the umami, right? It's just, it's so delicious. But there's so many other foods that have umami. I've actually started making my own umami paste. I found this little tube at Trader Joe's and I used it up and then it disappeared. So I had the ingredients. I made it all myself. Tomatoes high in the umami factor. Anchovies super high in umami. Let's see, black olives. That I mentioned tomato paste, mushrooms, balsamic vinegar. Mix all these things, grind them in the Cuisinart, and you've got your own umami paste that you can add, wherever you would want something that has more of a tomato base, right? So kind of like a sofrito in a way, but yeah, even glutamates, right? So MSG, it's controversial, but really high in that umami, that sixth sense kind of like, wow, it's so delicious, right? But in my mind, I'm gonna vote for the porcini. I don't know actual scientific data about it, but in my mind, the porcini is just like, it's so pungent and earthy. I would go with that.
I would guess the scientific data for a lot of people is going to come right from their tongue. So I'm hearing, yes, there's a range of umami flavors in the mushrooms, and which one you prefer is really going to depend on your own tastes. Morgan, I have one last question for you, unless anyone else in our audience has another question. And that is, I'm just curious, when we first started working together for forever ago, maybe 10 years ago now, you were doing programs just like this, cooking programs here at the Tilden Nature Area, and you drew these just huge crowds. I mean, and having watched you present over the years, I see why you're a fantastic presenter, and you obviously know your stuff about cooking. But my question is, how did you combine these two seemingly different niches of your love for food and the culinary arts and being an interpretive naturalist and a presenter?
Yeah, so I guess it just came naturally, foraging for my own foods. I did my externship at the Culinary Academy at Chez Panisse, and I was there during the winter months and basically was stuck in a dungeon cleaning giant sheet pans brimming with mushrooms. And so that's how I got involved in looking for my own mushrooms and foraging for my own wild edibles. And then at one point I decided, boy, I really need to get out of the food industry. This is just too much hard work. So then I said, well, I love being outdoors. So why don't I, look into a job in the park district? And having no idea what that was going to be like, I ended up here. I had great fortune to end up at the Tilden nature area. I think of this place as my second home. I'm very fond of it here. But I realized that people, like the common denominator people, no matter where you come from or what your politics are, is food. And so I would do a truffle program. I made goat cheese here with the milk from the goats on the farm. Everyone had to bring their own cracker. I had like 70 people in here wanting to taste the goat cheese. So just knowing that is something that brings people together. And of course, you want to talk about things you're enthusiastic about. So obviously for me, it's food. So, and the rest is history, I guess. But yeah, I love doing any sort of food programs that definitely, you want to up your numbers, add some food in there.
There you go. Food, a common denominator that unites us all. Well, Morgan, thanks again for joining us this year and showing us how you prepare this amazing dish. I know you prefer presenting to a real crowd, and we're going to make that happen next year at the 2023 Fungus Fair. So thanks so much for being here.
Thank you. Thank you. It was my pleasure. Thanks for having me, and I'll hopefully see you all around.
Sounds good. All right. Well, I don't know about you folks that are watching out there, but I'm kind of hungry after that. We have one final guest for the day, and I am really looking forward to this guest because he's giving a talk about a bioluminescence, and that is something that I find completely fascinating. Dr. Brian Perry is an associate professor of biology at Cal State East Bay. He received his master's degree from San Francisco State University in the Degrading lab and his PhD from Harvard University. Brian has been studying fungi since 1995 and has published over 40 papers in scientific journals. In addition to studying the evolution of fungal bioluminescence, he also conducts research on mushrooms and other fungi of Vanuatu, the assembly and biogeography of island fungal communities, endophytic fungi of Hawaiian plants, and the systematics of Mycena and allied genera. Brian teaches several mycology courses at Cal State East Bay and the Sierra Nevada Field Campus. Dr. Brian, thanks so much for joining us today.
It's my pleasure to be here, Trent. Thanks again for inviting me.
Oh, yeah, anytime. Now, if I remember correctly, you and I met at the SFSU Field Campus, which is just one of the most fantastic places in the world. I'm wondering, how is the field campus, and have you made it up there at all since the pandemic began?
Yeah, so as you know, there's been no courses up there for 2020 and 2021. We had hoped to have one last year, but had to cancel. But I still went up. So we've been collecting data up there for over 30 years now. So I did go up, stayed up there for about a week, just going to all of our normal sites, collecting, documenting everything that was there so that we wouldn't have any big gaps. As you know, we had some pretty hot, dry summers. So last June, when the course would have been going on, it was about 85 to 90 degrees up there. So not terribly conducive to fungal fruiting. So it's probably a good thing we didn't have the class. But this year things are looking better given the current snowpack. And the class, fingers crossed, is on the books. So registration actually opens tomorrow for the course.
Ooh, hey.
So for anyone that would be interested in coming up and taking the class, it's going to be June 5th through the 10th this year.
Okay, good to know. For anyone out there who's watching, I highly recommend this course. It's Fungi of the Sierra Nevada, and it's held at the SFSU Field Campus, which is just a fantastic place. So if you get the chance and you want to learn more about Sierra and fungi, take it. You won't regret it. I'm going to turn it over to Dr. Brian now for Illuminating Fungi, the Science of Fungal Bioluminescence.
All right. Well, thanks, Trent. Thanks for the introduction. I'm going to share my screen here. All right, so let's see. Okay, well, hopefully, can you folks see that? Give me a shout, Trent, if not.
Yeah,
There it
we can
goes.
see,
I've got you.
it.
Perfect. All right. Well, great. Well, thanks again for inviting me. As you saw earlier today, for those of you that were around, my student James presented, so it was really exciting to see him talking about his research. Fantastic, project that he's doing there in Tilden and some other areas. And that's just one aspect of what's going on in my laboratory. What I'm going to talk to you folks today about is fungal bioluminescence. And this is a phenomenon that I sort of got interested in and started researching almost completely on a whim. Back when I was doing my postdoctoral work with Dr. Dennis Desjardins over at San Francisco State University, we had a National Science Foundation grant to work on the genus Mycena. So the mycenoid fungi, if you know what those are, these little guys, sort of like the picture you see on the screen. And as part of that, he had just described a bioluminescent species from Brazil. Some folks down there had collected it and contacted him, and they had published it. And so when I came in to start my postdoc, we sort of got off on this tangent of looking into bioluminescent mushrooms. And it really just snowballed from there. It became this incredibly fascinating aspect of the project. We ended up forming wonderful collaborations with a number of international researchers. And now we're going on probably year 15 or 16, actually, looking at these fungi and working on them. And so what I want to share with you folks today is just tell you what I know about fungal bioluminescence and the mushrooms in which this phenomenon occurs, and then tell you sort of where we're at, what we've been able to discover about these organisms, as far as why they do it, how many times has it evolved, what is the biosynthetic pathway responsible for bioluminescence. So I'm going to tell you as much as I can about that. And I also want to say that, my co-authors on this talk, the folks that, have participated in the research that I'm going to be talking to you about today include Dr. Dennis Desjardins over at San Francisco State, and then my two colleagues from Brazil, from the University of Sao Paulo, Dr. Cassia Stovani and Dr. Anderson Alveira. So before we get too far into talking about bioluminescence, let's just first off, what is bioluminescence? And by definition, bioluminescence is really the production and emission of light in the form of photons by a living organism. And so what we're going to see today is that these are actually chemical reactions that are occurring within the cytoplasm of the cells of these organisms. And I want to make sure that folks don't confuse that with fluorescence. So fluorescence is going to be the absorbance of light energy and its immediate re-emittance at a lower energy wavelength. And I bring this up because in the sort of fungal world right now, there's a lot of work going on with fungal fluorescence. So especially a lot of the folks that like to go out and collect here in California, they will bring little UV flashlights with them. And when you're out in the field, just like we see with something like a scorpion, right? When you hit it with a UV flashlight, the outer tissues will fluoresce. So they're absorbing that light and they're immediately re-emitting it at a slightly different wavelength. So what you see in this photograph here on the screen is here you have a Russula species with this reddish cap, right? And this is basically a photograph taken when just a normal white light flashlight is pointed at it. And then over here on the right-hand side, you see what it looks like when you hit it with UV light. So the pigments that make up that outermost layer of tissue on that mushroom are absorbing those UV wavelength, that UV light of those wavelengths, and then re-emitting it at a lower wavelength or a different wavelength. And that's a very different process than bioluminescence. Okay, so bioluminescence is actually due to an actual chemical reaction that's occurring within the cells of these organisms. And what we're going to see, of course, is that bioluminescence is spread across the tree of life. So most of us are familiar with, made famous by fine movies like Finding Nemo. Here's an anglerfish, right? We know that anglerfish have these little appendages on their head. Inside of those appendages, they harbor a colony of bacteria. And those bacteria are bioluminescent. And we see something similar going on. This is in the middle here. This is a picture of a flask that contains bacteria that is bioluminescing. We know that in insects, right? So in our terrestrial systems, we have things like fireflies that produce little bursts of light when they're going through their nuptial flights and looking for mates. And then closer to home here in the southern Sierra Nevada, we actually have some millipedes that are bioluminescent. Okay, we have a few species that occur at different elevations in the southern Sierra. And these are some that were just recently described in not too long ago. And so this is a project I'm hoping to work on at some point in the future, is to go and actually collect some of these bioluminescent millipedes and look at them as well. In the marine systems, we have dinoflagellates, right? You can see these dinoflagellates tumbling in the surf here. I think this is Santa Barbara off on the left-hand side. And so these are being tumbled in the surf, and as they get disturbed and tumbled, they produce these flashes of light. Here we have a jellyfish, right? We know that many jellyfish are bioluminescent. We know that many fish have bioluminescent midlines or other appendages on them, like we see with the anglerfish. Some of that is due to harboring bacterial communities, and some of that light is actually just produced by the cells of the fish itself. And then down here on the bottom, these are glowworms. They're actually they're gnats that live in these caves in New Zealand, and what they do is they produce these slimy, sort of silky webs that they drape down from the roof of these caves, and then they're bioluminescent. So they produce light, and that light is to attract little prey species, which will get caught in that slimy filament, and then they can consume them as a food source. They basically reel them back up. It's almost like fishing with light in these caves. So we see it in both the marine systems and terrestrial systems, right? There's lots of bioluminescence, and it's even been reported that in marine systems, when you get down to the deeper depths where, sunlight doesn't penetrate, the only natural light that occurs at those depths is actually bioluminescence. It's being produced by fish and bacteria and a few other things, okay, maybe some algae, stuff like that. And if we look across the tree of life, what we see is that bioluminescence appears to have evolved independently over 40 different times, right? It's really crazy when you think about it. So here's the same sort of thing going on in all these different organisms, right? We have this light being produced by a chemical reaction in the cells, in the cytoplasm of their cells, but we see it occurring over and over and over again. And if you look at this is a phylogenetic tree here, so you can sort of think of it as like a family tree that represents evolutionary relationships and evolutionary time. You can see all these groups, and I know it's kind of hard from this figure, but this is a figure that I've taken out of a publication back from 2010 that was really focusing on bioluminescent organisms in marine systems. But you can see here, every single one of these lineages that has a colored font or a little bracket over here contains bioluminescent species. And so we see that in over 10,000 species from 800 genera across 13 phyla, this phenomenon occurs. Now, the phenomenon is distinct in all of these different lineages based on sort of the biochemical pathways, and I'll say more about that in just a minute, but it's also very similar, meaning that the chemical reaction that occurs in fireflies and fungi and dinoflagellates is actually very similar in the chemical reactions that occur, okay? So what we see is that these are distinct pathways, but they're also very similar, okay? And what this is telling us is that, wow, this phenomenon has evolved multiple times, probably for multiple reasons, right? And that leads us to a good question, and there's fungi, by the way, there. I've highlighted them in green. And a good question that I always get asked when I start talking about bioluminescence is, well, why do they do it? Especially when we think about mushrooms, why are mushrooms bioluminescing? And we'll come back to that at the end of the talk. What we can think about now is, in many of these organisms, we have a really great understanding of why they bioluminesce. So if we're thinking about fireflies, we know that they produce little bursts of lights when they're going through their nuptial flights and they're trying to attract mates. In the case of an anglerfish here, we know that this lure, this tissue that comes off of their head, harbors this community of bacteria that are bioluminescing. These live in deep, dark water environments, so they're using that light to attract prey species. Same thing going on here with these glowworms. They're using the light to attract prey species to them. And things like jellyfish, we might see that they're using light to attract prey, but they also might be using it as a form of warning coloration, where they're trying to discourage other organisms from eating them because they might be unpalatable. A similar thing might be going on with dinoflagellates. Dinoflagellates give this burst of light whenever they're disturbed, and that could sort of be like a burglar alarm or a startle response that's trying to confuse, predators that might be consuming them as a prey item. So there's lots of things and lots of hypotheses about why different organisms are bioluminescent. And we see that it really runs the gamut, right? There's lots of different reasons out there for why organisms are harboring bacterial populations that are bioluminescent, or whatever it might be. In the case of fungi, we still really don't know. Okay, so there's a lot that we have left to learn about why fungi are bioluminescent. But we've made really significant progress over the last 10 or 15 years in our understanding of why, at least why we think these fungi are luminescing. And I'll tell you about that in just a few minutes. Okay. So let's get into the fungi. Let's think about, what do we know about fungal bioluminescence? We can start way back with Aristotle. So we know, if we go back and we search the literature, sort of the first reports of fungal bioluminescence that we see go all the way back to Aristotle, right? And so Aristotle referred to really wood that was glowing with light. And he distinguished between this wood that was glowing, this greenish color, in comparison to, like, say, a piece of wood that had been burnt and was a glowing ember. So he distinguished that there was some sort of living light source in this wood. Okay, and that's really the earliest report that we have written down. Pliny the Elder, not the beer, but the man, who was basically a Roman philosopher. Pliny, in his writings, mentions a phosphorescent mushroom growing at the tops of trees in the Gallic provinces. So he's talking about bioluminescent species of mushrooms occurring in these European provinces. They weren't phosphorescent, of course. That's a different type of reaction. That's like a glow stick reaction. They were undoubtedly bioluminescent. Okay, and then we also have lots of writings throughout history. So Fernandez in the 1500s writes of Spanish soldiers in the West Indies that would use these little luminescent pieces of bark to basically make themselves visible to their comrades at night. And we see a similar report from Ramsbottom of soldiers using it in the trenches of World Wars I and II. And what they're talking about here is, if you've ever seen foxfire, if you know what that is, I'll show you a photo in just a moment. But a lot of times when you have a decaying log on the forest floor, if you start breaking it apart, you'll find big hunks of wood that are coated with the mycelium. Basically, you have mycelium that's breaking down that log, and it's the mycelium of a mushroom species. If you don't know what mycelium are, hyphae are these long filamentous cells. It's really the business end of a fungus. Mycelium is just a big mass of those hyphae. Okay, so if we think about a log being broken down by fungus, the mycelium of that fungus is going to be penetrating all throughout that log. And that's where it's exuding enzymes that are breaking down that dead wood. So in the case of something like foxfire, it's wood that's infected with the mycelium of a bioluminescent fungal species, and that wood glows. Okay, it's the mycelium that's basically coating that wood that is glowing. And so what we see is that soldiers would basically put little hunks of glowing wood on their helmets or someplace on their bodies so that they could see each other in the trenches. But the light is it's faint or weak enough that you can't see it from long distances away. So it'd be a safe way for them to essentially identify each other in the trenches. And then we see lots of other reports. So Olaus Magnus talks about using bark and basidiums in Scandinavia as a way to basically find your way in the dark, but also as a safe means of light to use in something like a barn full of hay, which can be highly combustible. Like going into a barn full of hay with a lantern or something could be incredibly dangerous, but going in there with a big hunk of glowing wood or some glowing mushrooms as your light source would be absolutely safe. Okay, and then no matter what culture you look at, where there is bioluminescence in that area, we see that there's oftentimes, reports of either ritualistic or practical uses from around the world. More close to home, we have one that is historical. So the Turtle submarine was the first American submersible that was basically built and used in combat during the American Revolution. So it was built in 1775, 76. And the goal of this submersible was to basically go out into harbors, bore holes in the hulls of the Royal British Navy ships and implant explosives and then blow up and sink those ships. So what you can see here, here's this diagram. These are these classic old paintings and drawings. Here's the man operating the submarine. Here's this little drill up here. And so the idea was that this would be basically a self-propelled submersible that could go out at night, go and drill holes into the bottoms of these ships and use that to try and sink them. Well, if you think about it, you have an operator locked inside of this submarine, and they need to be able to see the controls to know what they're doing. If you put a candle in there with them or a lantern, it's going to consume all of the oxygen pretty quickly, and the operator is going to very quickly run out of air and suffocate. However, if you put a hunk of bioluminescent wood, known as foxfire, into that chamber with the operator, it's going to produce light, but it's not going to consume oxygen at anywhere close to the same rate that something like a candle would. So again, it was a safe way to provide illumination inside of the submarine without consuming the oxygen that the operator needs to breathe, which is pretty darn cool when you think about it. And so the submarine was built. They used foxfire as a light source inside. I don't think it ever was successful in sinking any ships, and then eventually the ship that used to carry around the turtle was sunk. But there's a replica in some museum that you can go and check out. Pretty cool. All right. So that's one of the historical aspect of what we know about bioluminescent mushrooms. So what do we know currently? So what's our current understanding of these? So based on the work that we've been doing for the last 15 years or so, we know that there's over 100 species of fungi that have either mushrooms that are bioluminescent, or they have mycelia that are bioluminescent, or both that are bioluminescent. Okay, I think we're up to close to about 110 species that we've documented so far. And what we know about the chemical reaction is that it is well, basically like all other organisms in which bioluminescence occurs, it's a chemical reaction that is NADPH and molecular oxygen dependent, right? It's a reaction that's mediated by an enzyme that we generically call luciferase. And that's going to react with a substrate molecule or substrate compound that we refer to generically as a luciferin. And the end product of that is that in the presence of NADPH and oxygen, luciferase is going to basically catalyze a reaction where luciferin becomes oxyluciferin. The byproducts of that are going to be water. We'll see in a minute carbon dioxide, which isn't in this diagram, but most importantly is light. Okay, so we have this really basic reaction. What we know and what our studies indicate is that the luciferins and the luciferases are likely the same throughout all fungi that are bioluminescent. And this is really important, and this is one of the big findings that we've had from our research. Now, if we look at the actual chemical reaction, we know that it's very similar in nature to what occurs in something like fireflies. The firefly bioluminescent pathway is pretty well worked out. And in every organism that we've looked at, we know that we have an enzyme that's going to catalyze, an enzyme that we generically call luciferase. It's going to catalyze a reaction with some substrate molecule that we generically term a luciferin. What we see when we look at these different evolutionary groups, so whether we're looking at dinoflagellates or fireflies or bacteria, similar chemical reaction, but the compounds that form the luciferase and the luciferin are going to be different. Okay, and that's a really important distinction that we need to make between fungal bioluminescence and the bioluminescence that occurs in other organisms. Now, the other really cool thing about fungal bioluminescence, if you go and you read some of the old literature, you might see the reports of sort of like whitish or bluish light being produced by mushrooms, but we've never been able to verify any of that. For all the mushrooms that we have ever documented glowing, and all of those that we've measured, they all produce this greenish yellow light. And if you measure it with a luminometer, it's going to tell you that that light is in the 520 to 530 nanometer range. So right in that yellowish green spectrum. Okay, and they all glow at basically the same wavelength. What's also really unique about fungal bioluminescence is that unlike fireflies or dinoflagellates or other organisms, fungi are producing light 24 hours a day, 7 days a week. Okay, so in fireflies, we know they produce a burst of light. The same thing in dinoflagellates. They produce a burst of light during certain periods of their life cycle. In fungi, they produce it all the time. There's dips. Basically, it can be cyclic with regards to maybe there's more intense production of light during certain periods of the day, but they're doing it all the time. And in 2018, our Brazilian collaborators and some collaborators from Russia finally figured this out. So they finally proposed a model for the biosynthetic pathway that leads to fungal bioluminescence in mushrooms. And so for the chemists in the audience, what we see here is it's this really nice cyclic pathway where we have a substrate molecule that is being continually regenerated internally in the cell, such that as long as there is the presence of what you see here in this diagram, caffeic acid, we're going to have this reaction occurring continuously. So if we start up here and we look at caffeic acid, what we're going to see is there's just going to be a series of chemical reactions, which I'm not going to go into detail. They're all going to be catalyzed by different enzymes. Okay, and basically, you start with caffeic acid. It's going to be catalyzed into hispidin. Then hispidin is going to be hydroxylated into what we consider the luciferin substrate in fungal bioluminescence. That's 3-hydroxyhispidin is the molecule. And then luciferase, shown here as LUZ, right, another enzyme, in the presence of oxygen in that cell cytoplasm, is going to basically create a high energy intermediate molecule, which will quickly decompose, yielding oxyluciferin, carbon dioxide, and more importantly for us, light. Okay, that oxyluciferin can then be basically recycled by another enzyme back into caffeic acid. And so we have this really nice cyclic pathway where we can have both the substrate molecule, and the enzyme being continually regenerated so that these mushrooms can go through bioluminescence 24 hours a day, 7 days a week. Okay. And for those of you that have, found bioluminescent mushrooms, you know that you can pick them and you can bring them home, and they're going to continue to go through this process for hours after you've picked them. So basically, if you think of a mushroom as being, an apple, and the tree being the mycelium, even after you pick that apple, and take it away from the, the rest of the organism, this chemical reaction is still going to continue in the cells until basically the mushroom dries out or until there's no more caffeic acid present. One of these things is going to start to break down as that mushroom, decomposes and dries. But you can bring home these glowing mushrooms and they'll continue to glow for hours after you harvest them, which is really neat. It makes it much more easy to photograph them. All right, so we know a little bit about the history, how long we've known about fungal bioluminescence. We know about the chemical reactions there. I gave you a very quick sort of synopsis of what we do know about the biosynthetic pathway. And now finally, let's talk about, well, who are these bioluminescent fungi? So where do we see this phenomenon occurring across this fungal tree of life? And so it's really quite amazing. What we know is that all bioluminescent mushrooms are members of the Basidiomycota, right? And they're actually within the Agaricomycotina. And what this is telling you, for those of you that don't know all these names of these different groups within the kingdom fungi, it's telling us that they're all part of one evolutionary lineage within the mushroom, giant family tree. They're all white spored, right? So all of them produce spores that are either basically hyaline colorless. We call those white because they leave a white deposit when you put them on a piece of tinfoil or something like that. And in the old days, we used to treat all of these in a single family that we call the Tricholamataceae. Nowadays, that's been split up into a bunch of different groups, or what we call lineages. All of these species are saprotrophic, meaning they break down dead organic matter. And as we'll see in a little bit, all of them are actually saprotrophic, and they break down wood. With one exception, there's one species that's a parasite, but part of its life stage is bioluminescent. We'll talk about that one. And as my research has demonstrated, these occur in 4 distinct evolutionary lineages within the fungal tree of life. So we can look at that. And so again, what you're looking at here, this is a phylogenetic tree. It's just a big branching diagram that represents all the evolutionary relationships of all the organisms that are included there. And so what you see here, these are different groups. And I'm going to go through and talk about a few of these. So what this tree basically includes representatives from basically all of the Basidiomycota, right? So this big phylum here, we see all these groups in it. And if we want to know which ones are bioluminescent, well, that's what this next figure shows us. So here we see the Mycinaceae. This is the genus Mycena up here that I've shown in blue and some of its close relatives. And then we have these 3 other lineages, right? We have the one that we call here the Lucentipes clade, or the Lucentipes lineage. And then we have 2 down below here, one of which represents the genus Armillaria, and another one which represents the genus Omphalodus and some of its close relatives. So we have these 4 distinct evolutionary lineages in which we see bioluminescent species occurring. And we could think of this is being interpreted in 2 ways, right? We say, well, does that mean that bioluminescence has evolved 4 times independently, in kingdom fungi? Or does it mean that it evolved once early on and then was basically lost during evolution in many of the other species? Okay, and we'll come back and we'll talk about this in more detail in just a few minutes. So what we want to do now is let's get to looking at some actual pictures of these bioluminescent mushrooms, and I can tell you a little bit more about them. So first off, we'll start with looking at down here the Physolacranaceae and the Omphalodtaceae. We'll start with these 2, and then we'll work our way up this tree. Okay, so let's look at Omphalodus first. Omphalodus, the common name for these is the jack-o'-lantern mushroom. And there's 2 species I'm going to include here. There's Omphalodus olearius, which is a European taxon. We don't have that here in California or in the West. It's really common in Europe. Just like the Omphalodus species that we have here in California, it's toxic to mammals. So this is something you do not want to eat. And both the mushrooms and the mycelium are bioluminescent in these species. Okay, and we have the western counterpart to that species is Omphalodus olivascens. This is our western jack-o'-lantern mushroom. And this is really common throughout California and the Pacific Northwest. Okay, this is a mushroom species that was first discovered and described from material found in California. Just like its European relative, it's also toxic to mammals, and both the mushrooms and the mycelium glow. And for those of you that really want to see fungal bioluminescence locally, this is probably your best bet. And actually, Trent, our host, has this beautiful photograph that he made earlier this mushroom season. And so this is Omphalodus olivascens, and what you're seeing here, Trent did this beautiful image, combined both a shot in the dark and a shot in the light. So you can see the mushroom, and then you can just barely see the bioluminescence coming through. So you can see some green bioluminescence, sort of poking through. This is a really nice combined image. So probably the most beautiful shot of this species I've ever seen, Trent. So definitely well done on this one. Yeah, so for those of you that want to see bioluminescence locally, this is your best bet. Unfortunately, it can be incredibly variable in how bright the bioluminescence is. Some folks report being able to read a newspaper from, basically sitting next to this mushroom that produces so much light. Other folks report, basically having to lock yourself in a dark room for an hour before you can see any light being produced by these fruiting bodies. So it kind of depends on the age of the mushroom. Might depend on a lot of environmental features, like how moist is it out right now? Is the mushroom already drying out? Has it been rained on? Is it soggy? Lots of things can play a role in how bright the luminescence is going to be, but this is definitely your best bet to see a glowing mushroom locally. All right, and then a close relative of this is Neonothopanus, and this is not one that we have here in California. This is a photograph of material that I collected down in Micronesia on the island of Pohnpei, and the common name for this one is ghost ears. And it's a really amazing, incredibly bright, luminescent mushroom species. And it's called ghost ears because it reminds the locals when they go out into the forest at night and they see these mushrooms growing on the sides of trees. Well, they're kind of ear-shaped, right? They're kind of like an oyster mushroom shape, and they glow. And so they gave them the name ghost ears because they believe that the ghosts of their ancestors live in the forest. And so these are mushrooms that are known as ghost ears locally throughout certain areas of Micronesia. They're common both throughout the Paleo and the Neotropics, and we don't know if the mycelium is luminescent. We've never been able to get this thing in culture because when we were doing our research down in Micronesia, we simply didn't have the ability to culture mushrooms. We also don't know if it's poisonous. So we know that Omphalodus is toxic, but we don't know if Neonothopanus is toxic as well. We've never tried it, obviously, but we've never heard any reports of folks eating this mushroom. So we still don't know. But this is a really incredibly bright, really amazing mushroom to encounter out in the jungle. It's a really nice one. And then another species of Neonothopanus that's been really important for our research. This is Neonothopanus gardneri. And this is a species that was described back in 1840 from central Brazil as Agaricus gardneri, because everything was an Agaricus back in those days if it had gills. And it was first collected in 1840, and then it had never been collected again. So our collaborator, Cassio Stevani, who's down in Brazil, him and his students went out and they found this growing, basically growing at the base of these palms in this dry habitat just south of the Amazon. And brought it back. We worked with them on it, realized this was, this thing had been described back in the 1800s as Agaricus gardneri. They basically redescribed it and published it as Neonothopanus gardneri. And it's a really important mushroom for us because you can collect a lot of it. Okay, so this is the base of a palm here, and this gives you an idea of the size of this. This is a pretty good sized mushroom. And this is a really nice glowing cluster that was at the base of a palm. It's important for us because when the biochemists want to grind these mushrooms up and extract the luciferins and luciferase from them, you need a lot of material to grind up. This is a really important one because unlike many bioluminescent mushrooms that you'll see pictures of in just a few minutes, this thing is incredibly abundant. You can go out and collect pounds and pounds of it, right? So this is Dr. Cassio Stevani here. Here's a picture of him in the lab. And this is 4 2-gallon bags of dried Neonothopanus gardneri mushrooms. So a huge amount of material for the chemists to work with. And it also grows really well in culture. So this is one that you can either grow it on Petri plates, like you can see here in these photographs, or you can get bags of basically the palm leaves and you can sterilize those and then inoculate them with the fungus. And you can get these bags just filled with this luminescent mycelium that's really, really easy to maintain and easy to grow. So this fungus is really important to us because it really opened the door for us to delve into the biochemical pathway. It provided a sufficient amount of material for our biochemist collaborators to extract the luciferin substrate molecule as well as the luciferase enzyme that's catalyzing the reaction. So really important. I'll come back to that one in just a little bit. So that's it for the Omphaloctaceae. Now let's go look at the Physolacreaceae, because this is another one that we have locally. Okay, so Armillaria melia is a member of the Physolacreaceae. And as we know, it's really a complex of a number of species. So here in California, just in the Bay Area, We have a number of Armillaria species that we get growing here. So in the old days, we just sort of called all of them Armillaria mellia, but we now know there's probably a dozen different species hiding under that name. All of them are edible. So some of you have probably eaten these. You can go out and collect these big, huge clusters of them. People like to bring them back and they basically peel and eat the stems or the stipes, right? Some of the species are saprotrophs and some of them are root pathogens, right? So remember, I'm sure most of you have heard of the humongous fungus that we have in Michigan and Oregon. That's a species of Armillaria, right? So, and that's a root parasite species. So what we know is that some species seem to act more like saprotrophs, just breaking down dead organic matter, and other species are really pretty strong root pathogens on trees, okay? And as I said before, these are some of the largest organisms known on Earth, the humongous fungus, which covers these massive, massive areas. So that's Armillaria. Now, Armillaria is also what's responsible for foxfire. So Armillaria produces these really dense mycelial networks that we call rhizomorphs. These rhizomorphs become melanized. They're basically sort of like exploratory organs for the fungus. They're going to go out and they're going to explore the substrate looking for food resources. They can produce both rhizomorphs as well as just your common sort of, nice white, typical mycelium that's going to be breaking down that wood and growing all over. Armillaria species are what's responsible for foxfire. So here again is the same picture. This is a hunk of glowing wood, and it is a piece of wood that is completely infected with the mycelium of an Armillaria, and that mycelium is bioluminescent. Okay, so this is another one that you can see locally. I didn't think that we had foxfire very commonly here in California or in Western North America. It's very common in the Southeast. But over the last couple of years, we've been finding more and more foxfire by going out at night in the forest and basically finding a downed rotting log or a downed rotting tree trunk and just sort of pulling it apart and turning off your flashlight or your headlamp and letting your eyes adjust on a moonless night and seeing if you can find any of this rotting, glowing bark. So it does occur here. We do see it almost every year now. What's amazing about Armillaria is that the mushrooms that we have here in North America and in Europe are not bioluminescent. The mycelium of most of these species is bioluminescent, but the mushrooms are not. Okay, and that was always assumed to be the case, that no Armillarias were producing bioluminescent mushrooms, just bioluminescent mycelium. However, Taylor Lockwood was down in Australia photographing bioluminescent mushrooms, probably about like 8 or 9 years ago now. He collected Armillaria limonea, and sure enough, when he put it into his little dark cabinet that he created so he could photograph bioluminescent mushrooms, the mushrooms were bioluminescent. You can see it's just the cap. So here's the cap of the mushroom, but the gills and the stipe are not luminescent. It's just the cap, right? So that was really exciting for us because we had never seen an Armillaria species being documented as producing bioluminescent mushrooms. So really, really cool. And then right after that happened, our collaborator from Brazil sent us these photographs of this bioluminescent mushroom that him and his student had found and sent it to us. And we were looking at the photographs and we realized that it's an Armillaria species. And sure enough, this one also has bioluminescent mushrooms. Okay. So really, really amazing stuff. And just to give you an idea, in this photograph here that you're looking at, these species of Armillaria are tiny. They're probably, maybe 1.5 centimeters across the cap, right? That's compared to our local Armillaria species, which can be several inches across, right? So very, very different stature on these. But again, it's a new species we're currently in the process of describing from Brazil, from the Atlantic rainforest there. And it is bioluminescent. Both the mycelium and the mushrooms that are being produced are bioluminescent. It's really, really exciting for us. All right, so let's keep working our way up the tree. And next we're going to jump into what we're currently calling the Lucentipes clade here. And I have it in brackets because we haven't officially named this clade. It actually represents a new species, a new genus, at least one new genus. And we're working on it right now. So hopefully in the next year or so, we'll have a good paper out with a good name for these species. There's 2 mushrooms that occur in this Lucentipes clade. The first of them is Geronima viridilucens. And this is one that was described by Dennis Desjardins, Maria Capillari, and Cassius Stevani. This was something that was done right when I was just starting my postdoc at San Francisco State. They had just published this paper, and this is sort of what kicked off this whole sort of bioluminescence, road we've gone down. So This was the first report of a bioluminescence for the genus Geronima. And as I said before, it represents a new lineage of bioluminescence in the Euagarix, which is really another name for the mushroom-forming fungi of the Basidiomycota. It occurs on these specific trees in these calcareous soil areas of the primary Atlantic rainforest down in Brazil. And in this case, only the gills, only the lamellae are luminescent. The rest of the mushroom is not. Okay, so this one was really important because it was really the first one that kind of kicked us off. I can also tell you that it's not really a Geronima. So morphologically, it's very similar to other members of the genus Geronima, and that's what Dennis thought it was. But we know now that it actually represents a new genus. Okay, it's out there on its own. We finally sequenced its DNA and did some phylogenetic analyses. We determined that it's actually distinct. And the same thing goes for this other one. This is Mycena lucentipes, another really important one. These were published very close to one another in time. Another one published by Dennis Desjardins, Maria Capillari, and Cassia Stevani. And in this case, this is this beautiful mushroom that grows in these big clusters out in the Atlantic rainforest in Sao Paulo. And Dr. Desjardins was convinced that it was a Mycena, but it turns out when I was just starting my postdoc, I got hold of this material and I sequenced the DNA for it and I did a phylogenetic analysis. And I was able to show him basically the same week that this cover article came out that he had described it in the wrong genus. Okay, so it's not a Mycena at all. It actually represents an entirely new genus of mushrooms. So Mycena lucentipes and Geronima viridolucens represent two new genera of mushrooms, and it's an entirely new lineage of bioluminescent fungi that we discovered as part of our work. And what's even more cool about Mycena lucentipes is it's also the first and only, at this stage, bioluminescent mushroom to ever grace the front of a U.S. postal stamp. So for those of you that know Taylor Lockwood, he's a phenomenal mushroom photographer, has really gotten interested in bioluminescence over the last decade or so. It's been Taylor's dream to have one of his mushroom images on a stamp, and he finally got it. He actually submitted his photograph of Mycena lucentipes to the U.S. Postal Service, and it made it onto this whole series that they created called Bioluminescent Life. And so there it is, Mycena lucentipes on a postal stamp. This is pretty amazing. All right, and then finally, let's get up to the meat of this. This is Mycena. So this is the genus that I've been working on since I was actually an undergrad. When I was working with Dr. Desjardins at San Francisco State, he challenged me to do a monograph of the genus Mycena for California. For those of you that know what Mycenas are, they are basically small, little litter decomposing mushrooms that are really difficult to identify. So I spent about 4 years working on this group of mushrooms for California. When I was finishing up my PhD, Dr. Desjardins and I applied for and received a National Science Foundation grant to do basically a worldwide systematic sampling of Mycena. And so that's how I ended up working on bioluminescence, because I went there to work on that project, and we got basically down this sidetrack of bioluminescence because of that project. So looking at Mycena, lots and lots and lots of bioluminescent species. It's by far the largest lineage of bioluminescent mushrooms that we have in kingdom fungi. And it's also incredibly diverse. So you're going to see a lot of things in here that you might recognize, and you might not have realized that they were actually Mycenas. Okay, so this is Pinellus stypticus. This is another species that we have locally, but the species that we have locally tend to not be bioluminescent. Okay, so this is a really interesting species because when we look at the mushrooms and the mycelium of collections from Eastern North America, they're bioluminescent. When we look at collections from Europe, they are not bioluminescent. When we look at collections from the Western United States, they are not bioluminescent. So we have this situation where we have this large population. This mushroom basically spans the Northern Hemisphere, but only certain populations are bioluminescent. And so this is one that we're very intrigued to use so that we can basically compare the genomes of the luminescent and the non-luminescent populations and look for specific differences in there. So our hunch is that there's been mutations that have basically shut off the bioluminescent pathway in these populations that are not luminescent. And by sequencing the genome or the transcriptome, we should be able to figure that out. So this is one that we're currently working on, and we hope to do that with. And it's we do, I should say here, if you're in Eastern North America, you can find this mushroom bioluminescing out in the forest. You will encounter it here in California. I've never been able to find a collection that is bioluminescent. So if you do encounter one, by all means, please get in touch with me and let me know where you found it, because I would love to be able to find a local population that is bioluminescent. All right, then we got Mycena chlorophyos. This is probably the most widespread bioluminescent mushroom in the tropics. And let me hit the button here so you can see how it glows. It's really, really, it's a beautiful Mycena. It's very small, so the mushrooms you're looking at there, probably the caps are an inch or less in diameter, and the mushroom itself is probably an inch to an inch and a half tall. So they're pretty small, but incredibly abundant. So every place I've gone to do research, so whether we're working in Micronesia or other areas in the South Pacific, we always find this mushroom. Okay, so that's Mycena chlorophyos, really widespread, probably one of the more commonly encountered bioluminescent species in the tropics. Mycena luxcoli, again, this one looks very similar to some species we have here in California, but it's very distinct, both micromorphologically and in the fact that it is very brightly bioluminescent. Okay, so this one is common in the Asian tropics on decaying wood. Both the mushrooms and the mycelium glow. And what's really cool about this species is there's all these nocturnal spiders that hang out around it. And I think what they're doing, the reports claim that the spiders are eating the mushroom, but I think what they're doing is they're hanging out there and using the mushroom as a light source to attract prey items that we're going to come and check out the light. And then those mushrooms can basically, or sorry, the spiders can then capture and eat those prey items. So pretty amazing. Another really, really beautiful species. Hard for you folks to see it, but if you were able to see this thing up close, you'd see that the edges of the gills are colored, just like you might see in something like Mycena haematopus or Mycena californiensis or Mycena sanguinolenta here in California. This one has the same sorts of features, but is very distinct in other ways. All right, and then finally, we'll talk about one parasitic species. This is Mycena citricolor. Causes a disease known as American leaf spot on coffee. And it's really common throughout South America. I taught and worked in Hawaii for a number of years, and I was always curious to know if this fungus was ever going to make its way to the coffee plantations of Hawaii. I don't think it's ever been reported from there, but if it were, it would be another way that you could go out and see fungal bioluminescence. It's an interesting species. It's a really virulent pathogen, which is pretty rare for the genus Mycena. But what's amazing about this thing is it reproduces both sexually and asexually. And it's only the asexual reproductive structures that are bioluminescent. And so it produces these little structures that are called stilboids. And what these are is it's a way for the fungus to really easily infect an entire coffee plantation. These are basically asexual reproductive propagules. And here's an example. So here's a drawing of the mushroom that you can see. It's a few millimeters tall. And then down at the very bottom here is this tiny little structure. It's maybe like 2 millimeters tall. And that's this asexual phase. And it's just this little stalk. And on the top of it, it produces a little head. And that head is covered in sticky hairs. And that head disarticulates and falls off. And then it gets dispersed by water and wind, through rain and wind and things like that. And the stickiness allows it to stick to the leaves of other coffee plants and basically infect them and cause this disease. So those things are called stilboids. And it's this asexual phase that is bioluminescent. So if you were able to go out into a coffee plantation at night that was infected, you would probably be able to see all these glowing leaves because of the presence of these bioluminescent stilboids being produced all over the surface. And as was traditional in the old days in mycology, the asexual stage has a different name, so they call it Decapitatus flavidus, but in reality, it's just an asexual form of Mycena citricolor. All right, and then on to some more, just a few more, because I know everyone loves to see photographs. This is Phylloboletus manipularis. It's really a species of Mycena, based on all the phylogenetic analyses that we've done. Eventually, we'll be moving it back into the genus Mycena, so this will become Mycena manipularis. And it's, again, another really common species throughout the tropics, both the Asian tropics and the Pacific. And this is one that has recently made its way to Hawaii. So on the island of Kauai, a handful of years ago, a monk from a monastery who liked to do nature photography sent us photographs of this mushroom, just these big, beautiful clusters of it growing all over the property. And since that time, it's been documented a number of times in Hawaii. So this is one that has made its way north in the Pacific. We now have it in the Hawaiian Islands. So if you go on vacation to Hawaii, this is one that you might get a chance to see. It's a really incredible mushroom. It looks just like a normal mycena, but when you look at the underside of it, it has pores instead of gills, right? So this is one of these poroid mycenoid species. Really, really amazing. This is one of the first ones that I ever encountered in photographs. So again, this is work that I was doing down in Micronesia on the island of Pohnpei. And we collected this in the woods and brought it back to our apartment that we were living in while we were doing the research. And this is what it looks like when you turn off the lights. Okay, since then I've collected this thing all over the old world and the new world tropics. It's really common, and it's pretty amazing. You can collect a log like this, bring it back to your hotel room or your apartment, wherever you're staying, and this thing will glow all night long. Basically, if you don't, tear it off the log, if you leave that substrate there, it's going to continue to glow until the mushrooms basically dry out. So we use them as night lights. We'll put them on the, basically someplace in the room. We'll have these glowing mushrooms in our room all night while we sleep. So they're really, really fun, really amazing mushrooms. And then just a few more. This is Roridomyces. Here in California, we have Roridomyces rorida. You may have seen this one. It has this big, thick layer of snot on the stipe, right? And this is one that's it's fairly common if you're looking for little tiny mushrooms. But we also find these mushrooms all over the world, especially in the tropics. And the ones in the tropics tend to be bioluminescent. And so this is one that I connected in Vanuatu, and you can see just the base of the stipe is luminescent. So the whole mushroom, the gills, the cap, none of that is bioluminescent. It's just where the mushroom attaches to the substrate. And what that suggests to us is it's probably really the mycelium that is luminescent. And what we're seeing here is just where the mycelium is attaching the mushroom to the substrate. So we got really curious to know if Roridomyces rorida, which is quite common in California on the coast with conifer forests, whether or not this thing would be bioluminescent. So back in 2019, the last time we were able to have Soma Camp up in Occidental, if you folks are familiar with that, someone brought in a single tiny specimen of Mycena rorida, or Roridomyces rorida, as it's called, growing on an acorn. And Alan Rockefeller was up there and we were chatting about it. And I asked him if he should try to, I said, maybe you can go in and try and do a really, really long exposure so we can see if this thing is luminescent. And so Alan went in and found a dark room and put this thing in there and did a 220-second exposure with his lens all the way open, and the sensitivity on his camera turned up to maximum, and he was able to capture it. So what you're looking at here, this is the acorn, and then you have this little patch of green. And this little patch of green you see here is the bioluminescent mycelium of this species. So what's amazing about this is that our eyes cannot detect this. Okay, so human eyes aren't sensitive enough to see this bioluminescence. But if you do a really long timed exposure with a sensitive camera, or you put it into a luminometer, which is a machine that measures light output, you can actually prove or demonstrate that this species is indeed bioluminescent. Okay, and so this brings up a really important point about understanding bioluminescence, is that there's probably many species of fungi out there that are bioluminescent, but not at levels that human eyes can detect. And so one of the things that we do in my laboratory is we try and, every mycena that we collect, we try and bring it back to the laboratory and measure it in our luminometer just to see if it's putting out light at levels that our own eyes can't detect. Okay, so there's probably a lot more fungi out there than the 110 we currently know that actually are bioluminescent, but it's a really long process and you got to have the right tools in order to demonstrate this. All right, and just a few more just because they're cool. This is Mycena lux perpetua. So you'll see all these, these wonderful names. So the perpetual light is what that translates into. And this is one that is known from Puerto Rico, and both the mushrooms and the mycelium is luminescent. And this is one that I worked on back when I was a postdoc. This is one of the very first luminescent mushrooms that I got my hands on, was able to grow it in culture and get all these wonderful photographs. This is one that I collected and described from Borneo. This is Mycena sylvae-lucens, so the light of the tree is what that really means. And these are these beautiful Mycena species that grow on the sides of living trees in Malaysia and Borneo. Don't know if it occurs anyplace else. We've never seen it collected again, but the entire mushroom is bioluminescent. So really, really nice, beautiful fungus from Borneo there. Mycena lux eterna here. So again, the eternal light. And this is one that's known from Brazil. This is one of our favorites because it just has these incredibly bright stipes. So you get this wonderful clusters of these mushrooms growing on twigs and sticks and branches out in the Brazilian rainforest. It's really, really pretty. And then finally, I'll end with this one. This is a recent one that we collected in Vanuatu as part of my current research project. This is an undescribed species from down there. Really interesting mycena and shape and everything like that. But when you get it into a dark room and turn off the lights, you get this. So the stipes of these guys are really, really nice and luminescent. So again, another beautiful species that we'll be describing soon and naming it after one of our local collaborators and one of the local chiefs from the island where we collected this. All right. So that gives you an idea of how diverse sort of the bioluminescence is across the fungal tree of life. We have a large number of species that are bioluminescent. As I said before, we've got about 110 that have been documented so far. 70 to 75 of these species are all within that lineage that we refer to as the mycenoid fungi. So it's by far the largest and most diverse group of bioluminescent forming fungi out there. And so now we can get to the reason of, well, why are they glowing, right? So we spent all this time understanding, who it is, how long have we known this, what's the biochemical pathway, but we still don't really know why are they doing it. We know why fireflies do it, we know why anglerfish do it, but why do fungi bioluminesce? And there's a number of hypotheses that have been put forward. Probably the most common one is this idea that the fungi are bioluminescent so they can attract animals to aid in nocturnal spore dispersal. As we all know, by just turning on our lights outside during the summer, insects are attracted to light. So some of the original ideas were that by having glowing mushrooms, insects would come and visit those mushrooms. And by doing so, they would get either covered in the spores, or perhaps they'd eat the spores of the fungus, and then they would help disperse it, just like an insect helps disperse, helps pollinate flowers. The idea was it's going to be carrying away these reproductive propagules and spreading them around for the fungus. Sounds great, right? That seems like a really cool hypothesis, incredibly difficult to prove or measure, but it also doesn't work for species that don't have glowing mushrooms, right? And that's actually a lot. So we know that there's a ton of mycena species out there that do not have glowing mushrooms, but they do have glowing mycelia. So you can't really use that hypothesis to explain or to test rather what's going on there. It also wouldn't work for our parasitic species Mycena citricolor. Okay, so they attract predators of insects feeding on the mushrooms or mycelia. So the other hypothesis is that, well, maybe by glowing, it's going to attract other predators like the spiders that hang out near Mycena lux coli, and those insects are going to eat other insects that would otherwise feed on the mushroom. So it's a way for the fungus to protect its mushrooms and make sure they last as long as possible out in the environment. Well, again, interesting idea. Doesn't explain why the mycelium only of some is bioluminescent, but again, incredibly difficult to prove or to measure if this is really occurring. Okay, folks have proposed that maybe it's warning coloration, right? We see aposematic coloration in insects and other groups of animals, things like that. So perhaps this is something that's warning coloration being produced by the mushrooms. Well, probably not, because many insects still feed on toxic mushrooms and their mycelia, right? And then the majority of our luminescent species, with the exception of Omphalodus, are also non-toxic. So it doesn't make sense there. And that finally leaves us with what we're going with. So we're pretty convinced that fungal bioluminescence is simply an energy releasing, basically it's part of the metabolic pathway. And so what we think it is, it's a way for these fungi to release excess energy that they are generating as part of breaking down wood. Okay, so all of the mushrooms that we've been looking at, with the exception of the one pathogen, Mycena citricolor, everything else breaks down wood. In fungi that break down wood, we know that they generate a lot of things like what we call reactive oxygen species or peroxides. And part of those, if those build up in the cell cytoplasm, they can become very damaging. So we think that part of this might be, in order to neutralize those reactive oxygen species, the fungus is actually going through this whole biosynthetic pathway to neutralize those. And rather than release the energy as heat, like most organisms would do, they have simply evolved to release that excess energy as light. Okay, this seems to make the most sense, right? So we know that there's several compounds in that biochemical pathway to luminescence that are powerful antioxidants, so they could neutralize these reactive oxygen species. And all saprotrophic fungi require antioxidants for efficient litter and wood decomposition. So it seems to fit really well with what our understanding is of the ecological roles of these fungi. So we have a feeling it's really just a it's a metabolic byproduct of breaking down wood in these lineages, but that doesn't mean it hasn't been co-opted evolutionarily to do something else, right? So we might absolutely be able to prove someday that glowing somehow increases the fitness of these mushrooms and allows them to disperse their spores better by attracting insects. This is something that folks are working on right now. Some of our collaborators down in Brazil are making little fake mushrooms that glow at the same wavelength as fresh mushrooms, and they're actually looking at the insect groups that are attracted to them and how frequently they come and that sort of thing. But it's really, really difficult to prove that they're somehow dispersing the spores of the fungus and somehow increasing the fitness of that fungus such that natural selection would favor that process, right? But either way, it's an amazing phenomenon. So what do we really know about these guys? Well, there's 1.5 million of species conservatively, right? So we have lots and lots of estimates that go upwards of 5.5 million species of fungi. Less than, about 10,000 of those are mushroom-forming fungi, and the luminescent species represent about 1% of those. So they represent less than 0.005% of all fungi. So it's an incredibly rare phenomenon in fungi, but yet it's incredibly interesting to us. But again, an incredibly rare phenomenon. It occurs in 4 distinct evolutionary lineages, as we just looked at. They all luminesce at similar wavelengths, which suggests that there's probably a single origin of bioluminescence in the mushroom-forming fungal lineage. And as we'll see in just a few minutes, our previous work done back in the 1960s, and then our recent work published back in 2012, and then again in 2018, indicates that the luciferins and the luciferases that we see across all 4 lineages of bioluminescent mushrooms is the same, right? So this all suggests that we had a single origin of bioluminescence. And I'm not going to go into this in a whole lot of detail, but this is some of the early work we did. And what this is we're going through using all this molecular data. We build these phylogenetic trees, evolutionary trees, and then we use what's called character state reconstruction to predict what the probability of certain things is, right? So in this case, we wanted to know what was the probability that the ancestor to each of these groups was bioluminescent. And I won't go into this in detail. It's a horribly biased type of analysis that you can do that's completely biased by sampling. But what our initial data suggested was that, there is pretty high probabilities that the ancestor to each of these lineages, at least for the Lucentipes clade and the Mycinaceae, was bioluminescent. Same thing for the Physolacreaceae, not so much for Omphalotaceae, right? So, okay, interesting enough. What if we look at the ancestor to all mushroom-forming fungi down here? Well, we get this 65% probability that it was bioluminescent. So interesting, but not really, doesn't tell us a whole lot. Okay, 65% is sort of like, okay, that's interesting, but there's a lot more work we need to do. So what we decided to do was, working with our collaborators, was to look at the actual compounds responsible for the bioluminescent phenomenon. So working with Dr. Anderson Oliveira and Cassius Stevani. Anderson, when he was doing his postdoc, he came up to San Francisco State the same time I was there doing mine, and he worked on extracting luciferase and luciferin from all 4 different lineages, of these mushrooms, and then combining it. And so the idea here was that if you took the luciferase, which is the enzyme from, let's say, a mycena, and you combined it with the luciferin, which is a substrate, let's say, from Armillaria, if you mix those 2 things together and the bioluminescent reaction still occurred, that is evidence that it's the same bioluminescent system operating in both lineages. So what Anderson did when he was up at San Francisco State was he extracted luciferin and luciferase from samples of all 4 lineages. So here we have Armillaria, Neonothopanus gardneri, that one that we collected huge amounts of down in Brazil, Geronima viridescens, and Mycena luxaturna. And he would take basically you get the hot extract and the cold extract. So one extracts the luciferase and then the other extracts luciferin. And then we have a control here. This is Phylloboletus gracilis. This is a non-luminescent species that was the control. So when he mixed together the luciferin and the luciferase from all 4 of these bioluminescent species, the reaction occurred. Okay, and then when he mixed it with the non-luminescent species, there was no reaction whatsoever. So this was evidence that all 4 lineages have the same biosynthetic pathway right? Essentially it's the same luciferase, and it's the same luciferin operating in all 4 of those evolutionary lineages. So this was really, really great, right? Really exciting evidence that we have a single origin of bioluminescence in kingdom fungi, okay? And so our early analyses using basically just ancestral state reconstruction, and then the basically the much more robust analyses looking at the substrate and enzymes suggest that bioluminescence evolved only a single time in the Basidiomycota here, but that it was undoubtedly lost multiple times, right? So if you look at this tree and you see all these species here that are bioluminescent, so everything that has a little yellow dot is bioluminescent, but everything that doesn't is not bioluminescent. That means that thousands and thousands of species have lost this ability to bioluminesce that the ancestor to all mushroom-forming fungi likely had, which is pretty wild if you think about it. So that's where we're at right now. Okay, so that's our current understanding of fungal bioluminescence. We're actively working on this. This is a collaboration that has now been going for over a decade. And we're now at the stage where we're actually sequencing genomes of multiple bioluminescent species so that we can better understand everything that's going on with this biosynthetic pathway. All right, and with that, I want to say thank you all so much for listening. And also I need to actually acknowledge a lot of folks that contributed to this work, and also photographs from friends and colleagues and the National Science Foundation grants that have supported it. And with that, I will say thank you for listening.
All right. That was really fascinating. Thanks so much, Dr. Perry. Folks, if you have comments for Dr. Perry, put them in the comments sections now. Questions or comments, go ahead and type them in. There's a little bit of a lag before I can read them. Brian, when I reached out to you about this event, I was really hoping you'd talk about this subject because, as you probably know from our email exchange, I'm become somewhat fascinated with it since I finally observed our local bioluminescent Eucalyptus. Thanks for the love on that photo.
Of course.
And you mostly answered my first question, which was, why do you think fungi do this? So a follow-up question I have is there a way to determine if bioluminescence evolved first as a metabolic byproduct rather than for another reason?
Right. And yeah, so that's kind of what we're looking at right now. We'd like to know, sort of what's, can you actually make up that sort of decision based on the evidence we have? And right now we're just really trying to understand how common all the components of the whole biosynthetic pathway are. And it's really amazing. So what we'll actually see is that you can go and sequence the genome of, say, a close relative to, Armillaria or something like that. And it has most, but not all of the genes necessary for the whole biosynthetic pathway. So our collaborators in Russia, in conjunction with our collaborators from Brazil, published this paper back in 2018 that really, understood that biosynthetic pathway. Part of that was looking at all the genes responsible for the various enzymes that play a role. And what they're able to show is that it looks like those enzymes have basically been lost, either due to mutations, whatever it may be, in a huge number of groups. So we actually see the remnants of this historical ability to bioluminesce in many species that we've looked at. So whether or not from that we can determine, my feeling is it was the most logical explanation would be that it evolved first as a way to basically, as part of the metabolism so that it can break down wood, neutralize these reactive oxygen species, that sort of thing. So I think that makes the most sense of why it would have been selected for, under natural selection. But absolutely, I mean, if that occurred, and then it also increased the fitness of these organisms by, releasing energy as light rather than heat, because many of these things occur in tropical environments, and perhaps that's beneficial in some way, but also to attract potential spore dispersers. My feeling is that would be something that was secondary to this, more of a metabolic byproduct that evolved.
Okay, gotcha. Well, that definitely helps clear up some things. I've been reading a few different papers on the subject, and I was reading some of your collaborators' work down in South America about the internal timer or the circadian rhythms on Neonotha panis. Then also a study from 2016 that proposed that bioluminescence in Eucalyptus did not attract potential spore dispersing insects. So
Yeah.
yeah, it seems like a burning question in the bioluminescent fungal world.
Yeah, absolutely. And it's going, I think there was a nice study done back in the 60s that looked at this, and they were able to demonstrate that glowing mushrooms attracted more insects than did non-glowing mushrooms. They did this by putting them in glass tubes out in the jungle and painting the outside of the glass tubes with a sticky substance. And so this was like really the first time that work had been done. Since then, our collaborators have basically redone that study by making molds out of epoxy of mushrooms, and then putting an LED filament up inside of them that they can adjust the wavelength of the light with a rheostat, and they mimic, our glowing mushrooms out on these sites really, really well. So they can do multiple replicates of that, put it out into the jungle, and they've definitely demonstrated that the glowing mushrooms attract a lot more insects than do the non-glowing mushrooms. But that doesn't mean much, right? We still don't know what those insects are doing. So being able to prove that that's somehow beneficial to the fungus is really hard. You'd have to be able to show that those insects are actually dispersing the spores one way or another, or that they're eating things that would otherwise eat the mushrooms.
Right. And then you get into a whole different level where you're studying insects too. Sounds complicated. There
Yes.
are a few more questions coming in from the audience. Sarah asks, what species were the bioluminescent fungi in the intro slide? Were those mycenas in your first slide?
The title slide it's Bioluminescence manipularis.
Okay, there
So
you
it's
go.
the poroid mycena.
The poroid mycena, Bioluminescence. Krista, the Fungal forager, asks, could there be differences in gene expression that explain where they're within the same species that explain whether one is luminescent and another is not? Maybe something in the environment that turns it on or off.
Yeah, and that's where we're really curious. So we found a handful of populations or species where one population is luminescent and one is not. And so what we're trying to figure out, if we can get the transcriptome of those species, we can actually start to look, is either something missing or is the environment causing, basically the upregulation of certain genes, and that's what's causing the luminescence. So it's a great question, and we simply don't know at this stage. It's pretty neat when we go and we look across basically the fungal tree of life, and we look for all the genes that are responsible for that biosynthetic pathway. We see that many of them have been duplicated. We see that they have been lost. We see if they've been shuffled around. So, just like you would predict thinking about any genetic markers in a genome, they're also undergoing a lot of that. So you can imagine that, many of these mechanisms that we have that lead to genetic variation could be responsible for turning off that pathway. We also know that a number of the enzymes that are responsible actually arose more than likely due to gene duplication events historically. So that you got, basically genes that were, you had multiple copies, and it allowed them to, basically evolve into the pathway that we see today. But yeah, there's so much going on here that we have yet to be able to delve into, but there could certainly be environmental things going on that are impacting the expression of these pathways.
Interesting. Well, like so many things in science, more research is needed.
Yes.
All right, we've got your your crew is chiming in now with some questions. James asks, why do you think there is local bioluminescence on different parts of the mushroom or mycelium?
Could you say it again?
Well, why do you think there is localized bioluminescence on different parts of the mushroom or mycelium?
Good question. My feeling is it probably going back to gene expression, right? So if you think about what a mushroom is compared to the rest of the fungus, so the mycelium is where probably all those peroxides and other reactive oxygen species are being produced, right? Because that's really the business end of the fungus. So if we're thinking of these mushrooms breaking down wood, it's going to be in that mycelium where all those reactions are occurring. So you're going to have those enzymes there that are breaking down, cellulose and all that stuff, all that absorptive nutrition going on, whereas the mushroom is just the reproductive structure. So you can imagine that you would have the upregulation of genes in the mycelium, but there may not be the proper environmental cues to turn those on in these above ground fruiting structures. And so it might simply be that we see in some species they have the ability to turn that, those genes off in the mushroom, whereas in other species they simply don't. But yeah, good question, James.
Yeah, good one. From over on Facebook, Hart has a question.
Yes.
Let's see. Crotolobe and others suggested that this pathway could be encoded in most eukaryotes. Has this been done for any functional purposes?
Yeah, and it's really hard to say. So these are our collaborators from Russia are working with these folks, and it's really so we see that many of these genes we find them throughout eukaryotes. So as we see, like we've got that tree of life figure that I showed you. We know that bioluminescence is incredibly common throughout the tree of life. It's obviously evolved over 40 times independently in different lineages. So whether or not there's any functional purpose, whether or not we see it in multiple eukaryotes, who knows? I mean, I think a lot of the genes that are there are simply, we're going to find those genes, either paralogous copies, whatever it may be, orthologs in other eukaryotes, but they may or may not be, part of some pathway that we see in fungi. So good question, Hart, but I do not know the answer to that one. I'm not sure how far off we being from able to answer that.
All right. A somewhat related question. Just Jeff says, bioluminescence from bacteria is widely used in the sciences. Are you aware of particular applications for fungal bioluminescence specifically in other disciplines or industry?
Yeah, so it's actually our collaborators in Brazil have been using it for, like, basically when you inoculate stuff like sugarcane bagasse or palm stuffs, basically as fuel alternatives, what they're doing is they're basically taking this entire cassette of genes responsible for bioluminescence and they're transforming it into other things. So they now have a bioluminescent yeast, so a Saccharomyces species that is bioluminescent. If you can use that to inoculate things for biofuels, it lets you know if the fungus is present. So a lot of times when you're doing these inoculations, you don't know if your target fungus is actually successfully inoculated those. And so they're trying to come up with a strain that you can use for biofuel productions where it's really easy to determine based on the presence of luminescence whether or not you had a successful inoculation of the substrate. So, very, huge application there. And the really big one is natural light sources. So our collaborators in Russia with some folks from Europe basically have transformed this whole cassette of genes responsible for the pathway into tobacco, and they are growing bioluminescent plants. And so the idea is that you could grow, basically have natural free light being produced by plants. And then they're actually working with a company where they're trying to figure out how that can be adapted to produce lighting inside of homes. So these folks are hoping on big money coming out of this. So a lot of potential applications there, and there's a ton of it is already in work. Actually, I do have a slide in my talk. There's a really nice video on YouTube from these folks, and you can see all the tobacco plants that they've transformed to be bioluminescent. And so you have all these glowing tobacco plants growing.
Wow, that is fascinating. Yeah. I have to say, when you said bioluminescent yeast, all I could think of was, if I pitched that in my beer, I would know when primary fermentation is finished.
Exactly. I mean, so it's really their goal is to be able to transform this into a number of different things. We already use it a lot. So the firefly and the bacterial, biosynthetic pathways have been used extensively for a lot of different purposes. And so the fungal one is not too far away from being just as utilized, I think.
Oh, fascinating. Really interesting stuff. Couple more questions. Paul asks, do the same species that consume the gene bioluminescence also consume the non-bioluminescent?
Not sure what that means.
Maybe should have proofread that one first. Paul, if you want to reword that, we can try and ask it. I've got one more here from Dr. Puma. This is a bit of a softball. Can Dr. Perry give some advice on how I might strategize to see glowing mushrooms at their peak?
Yes, depends on where you live, right?
We'll assume here in California.
Glowing mushrooms. Yeah, if you're in California, I mean, kind of what I do is I go out during the day, and if I find, let's say I see a really nice Omphallotus olivascens fruiting someplace, I'll either, remember where it is, and then I will go back, assuming it's a moonless night, I will go back and try and observe it, in situ. If you're someplace where you can collect legally and you can bring those mushrooms home, then finding Omphallotus, bringing it home, and keeping it fresh, so that it remains moist and doesn't dry out or doesn't get too cold in the fridge, you've got a pretty good several hour window there where you can see it luminescing. I haven't had a tremendous amount of luck with our local Omphalodus. I mean, the photograph that you got is phenomenal, and that looked like it was a pretty intensely glowing specimen. I've talked to other folks that have found things right here in the Bay Area that you could read a newspaper by, from what they claim. So it's highly variable, and as I said before, I think it's really environment determined in many cases. So if it's too cold, like it's been now, we're getting down to freezing at night, things like that are going to really shut down the mushroom metabolism. And so that's going to lead to a reduction in bioluminescence. But I think that's probably your best bet locally, is to go out and look for that. As I said before, we're finding foxfire more and more frequently. I think just because more people know about this and they're out looking for it. So we were up at Soma Camp back in 2019, the last time it was offered, and a bunch of folks went out for a night hike and came back with all these glowing hunks of wood, that they had found, and they knew what to look for. So they went and they started tearing apart this old decaying log. And sure enough, there was foxfire there. So that should be one you can see as well. You can also grow it. So if you have the ability, if you're someone that grows mushrooms at home, you can get Omphalodus olivascens locally, and you can get it growing in culture. And as Hart discovered, oftentimes you have to get caffeic acid or grow it on a medium that's high in caffeic acid. So like barley or rye or some of these other common substrates for growing mushroom. And then you can get really nice glowing mycelium in culture. And that's kind of we do that a lot in the lab as we try and grow stuff, just because it's sort of a sure thing that we can show students and whatnot.
All right. Well, there you go. Grow it in a culture if you really want to be guaranteed that you see it. Also, for our viewers out there, I should note that photo that I took, that was actually a 10-minute exposure at a very high ISO. So that mushroom was way too dim for me to see with my naked eye. My camera could see it, but only after a long exposure. And related to that, we've got one more question here. It's kind of a two-parter, but has anyone looked at the effects? Michael asks, has anyone looked at the effects of light pollution on bioluminescent fungi? And could light pollution be used as any sort of a test to get at the function of bioluminescence in fungi? And I'll just say really quick that I struggled to take that Omphalodus photo because of light pollution. That was right here in Tilden Park. Yes. And on an overcast night, our sky just glows from the East Bay. So I actually had to hide the whole thing under a black umbrella when I was taking that photo. So, but that was just from a photographic standpoint. What about using it to look at the function of it?
Yeah, no, I don't think anyone, Michael, has looked into that. I don't know if there would be any effect. I mean, as I said before, these fungi are basically doing this on a 24/7 cycle. There's definitely this periodicity to it, so it's not, as you can read in the paper from our colleague, it does fluctuate throughout this 24-hour period, so you do get this circadian rhythm, but it's doing it all the time. So it's doing it all day long, and it's doing it all night long. So whether or not light pollution would really have any impact on it, I kind of doubt it. But it's as Trent says, though, I mean, it's really challenging here in the populated portion of the world to photograph these things. It's much easier when you're in the jungles of some island in the South Pacific where there's no electricity. I can tell you that much.
Yeah, I'll bet.
Yes.
Go to a dark forest on a moonless night. Yeah. Well,
yes, and Taylor Lockwood. Oh, we out.
Go ahead.
I was going to say, Taylor Lockwood has built these wonderful dark chambers. He does all of his bioluminescent photography in these boxes he builds. He puts them in the back of his car, and you can actually drag them out into the forest if he needs to. But he is actually doing this 360-degree rotation now on a turntable. So you can really see these phenomenal collections just spinning around and glowing. So a dark box is another option. A cooler. We've tried to photograph a lot of things inside a cooler. That's another handy dark container that you can use.
All pro tips from a professional bioluminescent researcher. Dr. Brian, thanks so much for joining us today. I am looking forward to getting out in the field with you at some point soon and hopefully finding some mushrooms. I thought we were going to get to do it this year, and it looks like we're going to have to kick the can down the road another year, but I hope we can make that happen in the not too distant future.
Me too.
Yeah. Folks, you can learn about Dr. Perry's current research at perrymycolab.com. I'm going to drop a bunch of links in the comments section in just a moment. Brian, thanks so much for joining us today.
My pleasure. Thanks for inviting me, Trent.
Well, folks, you've done it again. You've squandered an entire weekend watching live videos about mushrooms, mushroom people, mushroom food, and mushroom research. And now it's time to come to a close for the year. I'd like to give a big slimy mushroom thanks to all of our guest speakers for the weekend: Christian Schwarz, Dr. Gordon Walker, Dr. Monica Fisher, James Morris, Morgan Evans, and Dr. Brian Perry. I'd also like to give a shout out to our virtual vendors this year: Alison Pollack, macro photographer extraordinaire, and Coyote Brush Studios. You can check both of them out using the links on our website, tildenfungusfair.com, and I'll also drop their respective websites in the comments shortly. Special thanks to my co-host naturalist Anthony Fisher, who's been lurking in this feed for 2 days and helping me manage comments, levels, and buttons. And of course, a big thanks to my employer, the East Bay Regional Park District, for thinking this mushroom fair was a good idea 6 years ago when I proposed having our very first one, and still thinks it's a good idea today. And finally, thanks to you, the audience. You make this program, especially virtually, worth doing. So thanks so much for watching, everyone. I'm Naturalist Trent, signing off from the Tilden Fungus Fair. See you next year.